Hey, good morning. I just wanted to check if there's anything I should know about being on the Zoom. I'm not sure if I'm currently being live streamed to YouTube.
Hi, Rebecca. Can you hear me?
Uh, yes.
Uh, yeah,
Sorry, were you saying something?
Oh, I just wanted to come on a few minutes early because I'm going to be remote and check if there's anything I should know about.
Oh yeah, no, it's yeah, you just need to log in this way, and that's it.
yeah,
And
we're just want to check your video, Rebecca.
yeah, so when I click start video, it says unable to start because the host has stopped it.
Yeah, I'm gonna try it right now. Just give me a second.
And then the plan is to play my pre-recorded talk, and I'll be on for Q&A. And so if I'm just on in the background during my talk, then when the talk's over, I guess we'll turn on my video for Q&A. Is that how it'll work?
I think we're gonna play your pre-recording, and then afterwards, I'm probably just gonna step in and stop it and let people know that you're there live to answer questions.
Oh, so it won't even be video.
Sorry, what do you mean? Like you have the
Oh,
so
I
play that.
I just meant I'm not sure how it you'll feature my video during the Q&A. During the recording, it will just be a full screen of the recording, and then when it ends, you'll feature my video.
Yeah.
Okay, sounds good.
Yeah, let me just test it.
Yeah. Sure. Can you hear us?
Hello.
Sure.
Yes, I can hear you.
Oh, awesome. Okay, great. This is working. Thank you so much for figuring this all out.
And do we have a?
No problem.
Do we have a YouTube link?
Yeah, let me send it to you. I'll send it to you on the MLCB.
Oh, awesome. Okay.
I'll send you the link on the channel.
Hi, how are you?
Thanks for the invitation. Wow, loving the view.
Yeah. That's amazing. We're figuring out the speaker setups and stuff, but if I saw the Zoom, you got a Zoom panelist link this morning.
Rebecca,
So you can use your own computer, yeah.
Still
Yeah, that's
says,
another reason why doing it on your own laptop is nice.
Yeah.
Same thing. And just then.
Gerald, it
I think
still
it's
says
coming
I
from
can't.
Pleasure. How's it going?
Yeah.
Actually, a couple of people as expected dropped out, so it all worked out with the numbers.
Yeah.
Yeah.
As long as we can get the Zoom to work at least.
You were starting? Okay, you got the Zoom link.
And just then.
Just wanted to say thanks for letting me.
No, how's it going?
A couple of people as expected dropped out, so numbers.
Yeah.
Yeah.
As long as we can get the Zoom and everything to work.
You were starting? Okay, you got the Zoom link.
Sarah, I just posted the YouTube link in our Slack.
Okay,
Rebecca,
awesome.
can you just turn down the volume on
the website.
I just added to the website.
on your browser? Oh, awesome. There we go. Oh, I can't start. Oh, yeah, he needs to. Yeah. See if that works. Oh.
I don't think this is the, yeah, but you also need your sound options set in settings.
Sorry. Oh, the.
To go to settings, because you need the laptop. It's not just
Yeah.
laptop. You need QuickTime.
Let's see. Yep. Right. Okay. I think we're good.
Yes. It's either the small kind of family on.
Yep. think that's. Oh, do we have the video for this third speaker? 'Cause I don't have the link for them. Casio?
Should I still be standing by to figure out how to turn on my video?
Did you get it from me? Okay.
Should I send you an email?
Yeah, just send it my or yeah, whatever. The Kasha's
I just haven't.
I think maybe we'll give people a few more minutes. Yeah, sorry, I looked at I was reading the titles and I thought it was like something else.
Hey, I know that things are starting soon, so I just wanted to check. Should I still be on here trying to figure out how to turn on my video, or will we just do it without video? Now I'm not sure that you can hear me, which would also be a problem.
No, I can hear you, Rebecca.
Okay.
Sorry, I'm just trying to figure out some stuff with the webinar.
No problem. I'll be here if you want me to try again with the video.
Yeah, sounds good. Rebecca, can you try sharing your video again?
Sorry, what's that, Anshul?
No, I was trying to see if Rebecca can share her video. She says she's not able to. Not sure why, because she's a panelist.
Still not able to share my video. Maybe I can leave and
Can you
come
hear
back
me?
in.
Can you repeat that?
Oh, or Cassia just joined. Maybe she can try sharing her video, see if it.
Can you hear me now?
Yeah.
Okay, yeah, no, I was saying that I can hear Sarah very properly, but Gerald's pretty muffled.
Okay, so maybe is this like when I talk right now, you can.
Anshul, like, did you not hear the video when we were playing it?
I don't know what's going on. Where is that video sound coming from?
I'm so sorry. I had the live stream open. I thought that was today, like happening right now. I think it was yesterday's live stream.
Yeah, that was yesterday's live stream. Today's live stream is actually, I just put it in the chat. Yeah, but if you turn it on, it will definitely interfere with the live.
Guys, we're gonna get started, okay?
Yeah, you can get started.
Yeah. Okay. All right, everyone. Let's get started. So this morning we're going to start with our keynote speaker, Elham Azizi. So Elham is a professor of cancer data research and biomedical engineering at Columbia. Before that, she did her PhD in Boston University and then a postdoc with Donna Pierre, also Columbia, before she started her position a few years ago. And Elham is a tour de force, obviously at the intersection of machine learning, single cell genomics, and imaging for studying tumor microenvironments. And we're really happy that she was able to come and give us a talk. Take it away, Elham.
Gerald, we can't see video or screen. Are there slides being shared right now?
Do I just screen share? Oh, yeah, you just screen share.
Okay, great. Yeah, we can see the screen.
Okay, there you go. Anshul, can you see this?
Yeah, it's you need to swap your displays. It's showing the notes. Just swap the displays. Yeah.
Yep, you should be good.
No, it's still showing the wrong.
Thank you everyone for making it this early on a rainy Friday morning. I really appreciate it. And thanks to the organizers for this fantastic opportunity. It's really great to be back at MLCB in person. And MLCB has become one of the symbols of the comp bio field and how it's matured over the years. And looking at the poster session last night and your talks, I really see how far we've come in really blending and merging machine learning concepts with biology, and it makes me really excited about the future of the field. So first, to give some background, so a lot of our work is focused on dissecting the tumor microenvironment and specifically in specimens from patients, the patient samples. And because of that, because of the complexity of patient samples, we have to take, additional care and rigor in designing methods that are adapted to answering questions in this context. And the tumor microenvironment, just briefly, consists of this system of diverse tumor cells that are interacting with support cells, endothelial cells, fibroblasts, as well as a wide variety of if you see my mouse, maybe not, as well as a variety of immune cells, but I don't see it here on my screen, but that's okay. The immune cells that are infiltrating the tumor and try to eliminate the cancer cells. And so why is the tumor microenvironment important? We know now that there's a lot of great, strong evidence that the genetic alterations are not the only driver of response and outcome of patients, and the microenvironment also plays a significant role. And this heterogeneity of microenvironments across patients and within the tumors is important to dissect at multiple molecular layers, phenotypic, epigenetic, and so on. And so the problems that we focus on, and Bianca in her beautiful keynote yesterday highlighted and introduced these concepts of dynamics, but some of the major problems we focus on is using single cell resolution, multi-omics to understand the dynamics and altered cell state dynamics that happens in early stages of cancer initiation, as well as combining time series single cell data sets to look at temporal dynamics and how cell states change and evolve over time, in particular in response to treatments. In parallel, we also look at spatial dynamics and modeling how the spatial organization of cell types, diverse cell types in the patient tumor microenvironment also affects the response and outcome. And through this modeling of both temporal and spatial dynamics, we also want to derive principles about the interactions between diverse cells. And the major methods and models that we build are of the probabilistic flavor, and this audience does not need any convincing about the advantages, the theoretical guarantees, the interpretability, and the adaptability of these frameworks, especially for integrating multiple data modalities and ability to do hypothesis testing. So in the first part, I want to talk about some of our work on modeling spatial dynamics. This project was started with some of my previous postdoc work, where we were interested to know what's the impact of the tumor microenvironment in the diversity of immune cell states. So how do they change once they encounter the tumor cells? And in particular, we wanted to compare these diverse immune cells in the tumor microenvironment. How are they different from those found in the healthy tissue, especially from the same patient? And to do that, we looked at breast cancer patients and matched samples from the tumor, from healthy tissue, from bilateral mastectomies, as well as lymph node and peripheral blood, and sorted CD45 positive immune cells, and did single cell profiling and generated the first single cell breast immune atlas that revealed a diverse set of distinct cell states. And in particular, some of the important highlights of the study was that there's indeed a significant increase in the diversity of the immune cells when going from the healthy tissue. Once they encounter the tumors, we see a significant increase in all major cell types of T cells, myeloids, and NK cells. And for those interested, how did we quantify this diversity? We devised this metric called a phenotypic volume, which looks at the pseudo-determinant of the gene-gene covariance matrix. And so this intuitively, the geometrical interpretation is the volume that spans the eigenvectors characterizing the gene-gene co-expression matrix. And so if you have, for example, in the tumor, some higher numbers of co-expressed genes, modules of genes that are independent from those found in the healthy tissue, you expect additional eigenvectors and more independence of eigenvectors to be adding to this volume and showing this increase. So in addition to this increase in the diversity, one of the surprising results from the study was about the trajectory of T cell differentiation and activation. So if you look at the T cells going from the bottom of the arrow, from more naive central memory T cells, going to effector memory cytotoxic T cells, we expected that, just like textbook models of immunology, there should be distinct and defined states of differentiation along this axis, as well as activated T cells and exhausted T cells. But we were surprised that this distribution actually looks very continuous. If you look at, for example, the diffusion component, an axis explaining the variance across all T cells, you see that it's the least activated in blood and normal tissue as expected, highly activated in the lymph node, but in particular in the intratumoral T cells shown in the light blue on the right, you see a very continuous distribution. And so that was surprising for us. And we thought, could it be explained by the fact that there are so many antigen-specific T cells that were aggregating? Maybe the TCR, the T cell receptor profiling of that can help deconvolve them. So we did pair TCR and single cell, and the answer was no, that the T cell clonotype identity actually doesn't fully explain this continuity. When we looked at defining, rough clusters, even though they're not distinct, and looking at the expression of signatures shown here in rows of these heat maps related to responses to microenvironmental stimuli, we see that the clusters show different combinations of levels of expression of representing responses to these stimuli. So the hypothesis that we took away from this was that maybe we have T cells exposed to different niches in the tumor microenvironment that are also differing in the levels of oxygen, nutrient supply, it's activating ligands and cytokines and so on. And because of that, we're looking at the aggregated from dissociated tissues in single cell profiling and therefore seeing a continuous distribution. So the answer was, okay, let's do spatial. Now we have spatially resolved technologies that can, resolve the phenotypes in the spatial context of the tissue. And again, thanks to Bianca for introducing spatial methods. We continued our collaboration with Sasha Rudensky and George Politis at Sloan Kettering to do Visium spatial transcriptomic profiling of breast tumor tissues and to try to understand if the spatial organization defines immune differentiation and stromal response. We were also particularly interested to compare spatial organizations across tissues. For example, in metaplastic breast cancers, these are a type of triple negative breast cancers that are more aggressive. They actually grow on chemotherapy, unlike other triple negatives, and they have different morphologies, very heterogeneous morphology. If you can see on the histology slides, some of them look like soft tissue and some connective tissue. And so there's a lot of demand tailored for this very aggressive subtype that, is adapted to the unique biology of this breast cancer. But to be able to compare spatial organization of cells across samples, we need new methods as well. Also, in addition to that, as many of you know, the spatial transcriptomic technologies, which look at sampling the entire transcriptome, are limited in their resolution. They're not exactly single cells. So you have a mix of multiple cells within every spot shown here in the zoom out plot here on the top left corner. And so this is a problem when you're looking at complex patient samples because you have mixing of immune cells that you're trying to compare across tissues with patient-specific tumor cells. And because they're patient-specific, there's not going to be any overlap between spots. And in particular, if we want to really deconvolve the refined cell states like activated T cell states from exhausted T cell states, we need methods that are able to deconvolve spots at that resolution. And when looking at there are many great methods out there now for deconvolving spatial transcriptomic data. The majority of those that are able to get these refined cell states rely on a single cell reference that is from the same match sample. And this has its limitations, but also it's, something that might be limiting when you're looking at patient samples and you might not have enough material. So a number of brilliant students in my lab, CU, Yino, and Ashil, and also my postdoc LinkedIn, worked a lot on the interpretation, thought together about how can we address these issues to deconvolve cell states without the need for a single cell reference, and also leverage all this information that we have on the histology imaging. This is what the pathology uses for the annotation and the diagnosis. So it has a lot of information about the density of the cells, the tissue architecture. So why not integrate these two data modalities to improve our understanding of the spatial dynamics? And most importantly, we wanted to build a framework that is able to do comparative analysis of multiple tissues. So this led to Starfish. I'm not going to read the full name. I leave the naming of tools to my students. So they come up with these very long acronyms. But the idea is to combine spatial transcriptomics and histology. So to give an intuition on how we avoid the need for single cell references, I'm going to show a cartoon that is, simulated data that is generated based on mixtures from a single cell RNA sequencing data with known mixture levels from a breast cancer tumor data set with 5 major cell types, CAFs or fibroblasts, tumor cells, myeloids, normal, and T cells. And you can see the UMAP Just like the UMAP of any spatial transcriptomic data doesn't show defined clusters of cell types because you have these mixtures, but at the corners you see those are spots that are enriched for markers of known markers of cell types. Now, we thought, okay, if a user has knowledge of the markers of these cell types, can we use those as guidance for the deconvolution? But in many cases, especially in patient-specific tumors, you don't have accurate markers for those cell types. And so the idea we used is the concept of archetypal analysis, basically fitting a polytope to the low-dimensional representation of the data, and the extremities, the vertices of this polytope are the extreme states that the spots can reach. And our major assumption is that those extreme points represent the purest spots in our data, meaning they're either consistent of one cell type or dominant in mainly one cell type. And we recently tested this assumption thanks to our reviewer as well on Xenium single cell resolution data, and indeed it is a correct assumption. And then finally, we align this, if there's information about the known markers, we align the archetypes to kind of adapt our marker set, any known marker sets, to the context of this tissue, and then use these anchors to kind of pull apart the rest of the spots. We get these starfish-like shapes, hence the name, and then we show with this data that it fits the data. For the more technical details, what exactly do? We have, so X here is our transcriptomic data, expression of spot I in gene G. We have a variational autoencoder mapping it to a latent space Z. And Z is centered around a weighted sum of our anchors, which we also learn, but we in this pre-processing step, they are guided by the definition of the archetypes. And then C here gives you the proportions of the cell types, which you also learn. Theta is just the gene dispersion, L is library size, similar to other deep generative models. And finally, we have this additional data modality, the histology image, as a second autoencoder, but a mapping from the same joint latent space. And again, Bianca mentioned the advantages and benefits of learning joint representations from multiple data modalities. We indeed see that it helps with improving the deconvolution of tumor cells and fibroblasts using this integration. So we show on simulated data, we do as good as methods that use a single cell reference, even without using it, but significantly improve over methods that don't. This is now has been expanded to higher numbers of cell types. We use data set recently matched Xenium, so single cell resolution, but lower dimensional, and Visium data from the same, from adjacent slices of the tissue to actually evaluate, are we getting the correct deconvolution? So here you can see two types of invasive tumor cells at the bottom and non-invasive ductal carcinoma in situ cells on the top. And you can see it does align with the inferred proportions from the Visium data. But what was really interesting was that we found the archetypes actually were able to deconvolve kind of in a hierarchical manner, further distinguish two different subsets of these non-invasive DCIS cells shown at the far right there that are exactly aligned with expert annotated regions shown in the red and yellow in the histology. And so without any knowledge about what are the markers that disentangle these two more refined subsets, we found archetypes that exactly match them. So what do we learn about the spatial dynamics? So here, this is an example of a triple negative breast cancer tumor. First, we define spatial hubs or neighborhoods by grouping spots that have similar inferred composition of cell types. And so now we can look across these spots that are high in tumor cells, define trajectories of how the phenotypes change, are changing and evolving across these spots. And we can see, for example, going from the cyan to the higher top left part of the tissue, we have a reduction in cell adhesion molecules, an increase in mesenchymal markers and EMT. So really showing that their cells, the tumor cells, are changing from more basal states to more mesenchymal states within the same tumor tissue from a patient. So now, how are the immune cells changing if the tumor cells are adapting and changing in phenotype? We see that, interestingly, as there's this shift in the tumor states, we also have a shift from more central memory to effector memory cells, and then activated CD8 T cells at the margin of the tumor, and also shifts from monocytes to macrophages. So this really kind of goes back to the hypothesis that we started with, that there are indeed different immune cells associated with different niches of the tumor microenvironment, and the tumor cell they're interacting with actually does impact or determine the immune cell phenotype as well in the same spatial neighborhood. So in addition to this integration of histology and spatial transcriptomics without requiring a reference, Starfish is also able to integrate multiple tissues and create these atlases of spatial hubs or neighborhoods across multiple tissues, and then do downstream analyses like just what I showed, this pseudospace, or how phenotypes change across the space of the tissue, and then do cell-cell interaction prediction. So this is an example of an integrated space of 14 samples, 14 breast tumor samples, triple negatives and metaplastics. You can see the UMAP is just showing clusters that are patient specific, no overlap, despite the fact that there's common immune cells among them, whereas Starfish is showing a merged and blended and mixed latent space with more mixing in the immune cells like Tregs, and less mixing in tumor enriched spots. Also quantified with entropy, and then you can also see the space colored by the spatial hubs. So now with these spatial neighborhoods defined, we can see what type of neighborhoods are, for example, enriched in the metaplastic breast tumors shown as MBC here. For example, hub 0 is one case, or what are the types of hubs that are common across all breast tumors. And just as sanity check, we also compared to, expert annotated histologies. We do see that, for example, that blue hub there in the middle is aligned with a healthy annotated tissue from a pathologist. Also, between two adjacent slices at the bottom in the middle, we have reproducible hubs identified, showing it works on replicates and gives same results. But most importantly, we can compare across samples. So how do the tumors, how are the spots in metaplastic breast tumors in the intratumoral regions compared to the triple negative? So looking again at this joint latent space, we find that there's an increase in inflammatory response, but more interestingly, in immunosuppressive cell types, such as regulatory T cells and myeloid-derived suppressor cells, or MDSCs, in the metaplastic breast tumors. In addition to that, we also see an increase in perivascular-like cells, which might indicate angiogenesis. And we also see interesting stromal hubs in the metaplastic tumors with a very concentrated organization shown in these red pockets across the metaplastics that are also exactly overlapping with the regions that are high in hypoxia. So low oxygen levels regions are forming these regions that are high in immunosuppressive cell types like regulatory T cells, and we also see an increase in perivascular-like cells. So we think that the regulatory T cells might be contributing to building these immunosuppressive niches and potentially adapting by angiogenesis. And if you zoom in the histology, we also see some red blood cells forming around the region. So there's an interesting connection with the immune cells. We validated these immunosuppressive niches recently with Codex data profiling based on markers that we found in our transcriptomic level deconvolution. And lastly, we looked into predictions. Can we predict from the deconvolved gene expression profiles what might be the receptor ligands that mediate interaction between the regulatory T cells and the tumors? And FGF2 pathway was one that is coming up a lot and does show this decline by distance only in the metaplastics and not in the triple negatives and ERs. So I want to wrap up this first part. I showed Starfish, a deep generative model that uses anchors guided by archetypes, that replaces the single cell reference, does image integration and comparative analysis of tissues. And I showed some results about insights we get in comparing metaplastics and the enriched immunosuppressive niches. I want to move on now to temporal dynamics as another perspective to look into cell-cell interactions, and in particular, longitudinal profiling, profiling of tissues at multiple time points, for example, before and after immunotherapy, which can tell us how are the immune cells changing once the patient is given immunotherapy in the tumor microenvironment, which ones are infiltrating, which ones are dysfunctional in the patients that are not responding to the treatment, and how can we reverse the immune dysfunction. So in our paper published earlier this year in collaboration with Ben Iser, we looked at anti-PD-1 therapy treated melanoma patients, and looking at single cell RNA sequencing at multiple time points before and after, we see, for example, cases like this red clone defined from INFER-CMV that is shrinking and responding to the treatment. So it disappears in the on therapy samples. But we also see cases like this purple clone that you see it expands in proportion. It's seen at very small proportions pre-therapy and significantly expands. So, and this patient was a non-responder. So there are, some clones in the tumor microenvironment that are resistant, and we wanted to know, is there any difference in terms of the spatial organization of the immune cells around these resistant clones compared to the responsive clones? And indeed, applying Starfish to slide-seq data now instead of Visium, we were able to disentangle activated from dysfunctional CD8 T cells, and saw that the activated CD8 T cells are only co-localized with the responsive tumor clones, whereas we see dysfunctional T cell clones, T cells co-localized with the resistant clones. And so this level of disentangling of T cells was not possible again with other methods, for example, RCTD methods that don't rely on single cell resolution. In other cancer contexts, for example, in leukemia, we try to draw principles about response and resistance to immunotherapies from established types of therapies. For example, donor lymphocyte infusion is a adoptive cell therapy that's given to leukemia patients that relapse after stem cell transplant. And we don't know exactly how they work. They used to work very effectively for chronic myelogenous leukemia patients before imatinib and targeted treatments. Today, they are the standard of care for AML patients, acute myeloid leukemia patients that relapse, but they don't work as well. They work up to 20, 30% of patients. And so we wanted to see, can we draw, principles about DLI response from CML cases, CML patients, and use that to guide how we can improve treatments for AML. And in my previous postdoc work, we focused first on CML, and using Gaussian process regression models that I won't talk about today, we uncovered two major subsets of T cells that are, in particular, this dark blue cell types that light blue cell types that are exhausted but also have stem-like properties, and they expand after treatment, and we think they are shaping the response. And we wanted to see if they're coming from the infusion, the therapy itself, or is the therapy reinvigorating interactions among immune cells in the tumor microenvironment using T cell receptor data. And our data was suggesting that it's rather the latter, and it's invigorating interactions across cell types. So that really motivated looking and trying to model interactions between cell types in the tumor microenvironment from this longitudinal data. So we started this collaboration with Kathy Wu at Dana-Farber, Katie, a postdoc in her lab, Cameron, PhD student in my lab, leading this project with Katie, and also Shovik Mani, who was a master's student in CS in my lab, and now a PhD student at Stanford. We worked on this project, ambitious project of trying to compare two different cohorts, AML and CML, to try to understand how their DLI response is different. And so just creating this atlas of more than 400,000 cells, we find there's a lot of indeed overlapping immune populations, while there's distinct diverging leukemia trajectories. But to get at cell-cell interactions, you've probably heard of many interaction prediction methods, NicheNet, CellChat, CellPhoneDB. They essentially look at differential expression of receptor ligands in clusters of single cells. And You might kind of even talking to our biologists, it's really hard to prioritize which of these many hits predicted receptor ligand interactions might be relevant to the context of this disease. And so our idea was, can we actually learn from the temporal dynamics of cell types, which cell types might be interacting, and at least combine that with prior knowledge. For example, if we see a decline in tumor cells and increase in a T cell, maybe the T cell is inhibiting the tumor cell. So that negative temporal correlation is informative about potential interactions. So the idea of DISCO, dynamic intercellular interactions in single cell transcriptomics, is to exactly combine these temporal correlations with prior knowledge of receptor ligands to try to constrain the space of possible cell-cell interactions, but also get time-resolved interaction predictions, because obviously after therapy, we expect a different set of cell-cell interactions in the tumor microenvironment. The idea of the method is actually building on beautiful previous work from David and others using Gaussian process regression network models, where we model every cell type as a Gaussian process shown in Y, and we have latent features F1 to Fk for K cell types, but we also have this W parameter here, which is also a Gaussian process showing the interactions between cell types. And so this model by itself is not identifiable. So our idea was to constrain W's using this prior knowledge of receptor ligands to have, improve the identifiability and also get time-resolved interactions. So this is now public as well. Feel free to use it. If you have time series single cell data, let us know how it works. To show you, in our preprint, we show the performance on both simulated and also more controlled experiments, in vitro experiments with CAR-T and cell line interactions. But here I want to show some exciting results on the full AML cohort. We see that there's different types of T cells from what we found in CML, actually potentially driving the interactions, more CD8 TEMRA cells that, in addition to NK cells and other immune cells such as B cells, that are also expanding in proportion in AML responders and inhibiting the proportions of leukemia. So these lines here, the line is showing the mean of the Gaussian process that we fit to the data points, and the confidence intervals are shown in shaded areas. But we also learn about interactions, and we can have time-resolved interactions, which cell types are interacting. For example, after therapy, we see these immune cell types, the CD8 TEMRA, NK cells, increasing in a proportion and strongly interacting to inhibit the leukemia cells. And this cool video by Shovik, if I can get it to run, also shows how the proportions of cell types and the interactions change, so you can have predictions from the Gaussian process at any time point. So next one. Now it doesn't stop playing the video. Okay, there we go. So we're now looking into validating these interactions using spatial codex data that I won't talk about today. But just to recap this second part, DISCO is able to infer time-resolved cell-cell interactions, and in our AML patient cohort, it is identifying coordinated immune networks that are not found in more homogeneous leukemias such as CML, and this was possible with longitudinal single-cell mapping. All right, so if we have a couple of minutes, I want to talk about another exciting project now on the dynamics of cancer initiation, keeping the best, saving the best for the last. So this project is trying to get at, started with answering this question about tumor initiation. So we know that, for example, chemotherapy, a lot of standard therapies eliminate the most mature tumor cells. And these early precancerous cells that have stem-like properties are able to grow back, for example, leading to relapse and metastasis. And we don't have very good markers for them in the majority of cancer types because they are rare and hard to profile and characterize. So our question was, can we use, trajectory inference methods and single cell resolution profiling to try to reconstruct this tumor initiation trajectories? And we focused on AML. So AML, bit more background. Normal hematopoiesis is shown on the left. You have these hematopoietic stem cells at the root of the tree, differentiating into progenitor cell types, and then differentiating into all your white, red blood cells continuously. And in the context of AML, we have a block differentiation on the myeloid arm, and we think what's happening is that the cells that are either stem or progenitor lead to precancerous cells, and then through acquisition of accumulation of mutations and alterations, they lead to leukemia cells. So towards the end of my postdoc, we collected some data with Vincent Lavallee, who now has his own lab at the University of Montreal, and Donna, looking at multiple patients, 12 AML patients, that we tried to focus on particular types of mutations, TET2 epigenetic mutations and NPM1 driver mutations, to at least have less heterogeneity. This is a very heterogeneous cancer type. And again, we see that all of the main clusters, major clusters, are patient-specific. There is some overlap between patients in this lower left populations here in the T cells and B cells, as expected, because this is a myeloid malignancy. The lymphocytes should not be affected. But what was surprising was the circled population here, this immature population, also the Shannon diversity, quantifies the high mixing of multiple patients. So it's a population that's highly correlated with healthy hematopoietic stem cells, but found in 11 out of 12 patients. So that was interesting. We thought maybe this cluster includes some of the precancerous cells. And so we found markers for them, zoomed into this population by sorting for them, doing additional round of single cell profiling. And we saw that it consists of two major populations, one more stem-like and one more progenitor-like. And what was really cool was that when scanning for the mutation of the driver NPM1 mutation, which happens on the 3' end, that's why we chose this mutation, and leads to high expression, so we could detect it from single-cell RNA sequencing 3' data. We saw that the mutation exists only on the progenitor-like cells and not on the stem-like cells. So this really kind of suggested that there's a direction of mutation events accumulating, and we really wanted to study what are the transcriptional consequences of these mutations along the full trajectory of tumor formation. So for that, we thought, okay, how about comparing these cells to healthy hematopoietic cells? And that turned out to be challenging when you compare these now sorted cells in purple on the left with a healthy CD34 positive hematopoietic stem cells in green. You see that integration methods that basically don't show any overlap or any idea of the global geometry. We also have this annotation of the maturation stages of the leukemia cells from blast 1, blast 0, blast 1 to 3. We see that the ordering of these cell types doesn't make sense in any of these representations. It also doesn't make sense when we try to do batch correction, because batch correction is designed for correcting technical artifacts between samples. So if you're comparing disease samples where the majority of the cell types are not found in the healthy tissue, it's just trying to force them to overlap. So these methods shown on at the bottom row are really trying to force the cells to overlap on top of each other, but they might be misleading in the interpretation. They can tell you, for example, in Surat, that maybe it's the healthy cells branching and bifurcating to the blast 2 and blast 3, which are the more mature leukemia cells. So obviously incorrect representation of the trajectory. So two brilliant, talented students, Shiel and Joy in the lab, thought of designing a new method that tries to address these issues. So essentially what we thought about is, if we're trying to get an interpretable representation from two data sets, for example, normal and a perturbed or disease data set, there must be shared mechanisms between these two trajectories. For example, cell differentiation is a preserved mechanism between healthy hematopoiesis and leukemia. But they might also be diverging, so leading to very distinct cell states. So we thought, can we relax the assumption of independent and orthogonal latent components in our representation? And so this, it becomes challenging, again, dealing with identifiability. But the idea we came up, and I'll show the details in a second, indeed shows that if you allow the factors, the latent factors, in this case, deep generative model, to have correlations between them, they can exactly resolve and disentangle these cell types shown on the right that are distinct, whereas other VAE methods like SCVI completely overlap these cell types and do not distinguish them. So how does the method actually work? What we have here is, really building on methods like SCVI. So we have this VAE mapping X to Z, but the major novelty here is adding an additional layer where the latent factor Z are actually a function of another layer V, which is even lower dimensional, in our case, a two-dimensional variable. And so there are multiple advantages of this defining as a 2D space. We can directly use this model to visualize our data in a meaningful manner and not have to do UMAP or another visualization method that distorts the geometry of the data. And also we have this Z factors that are defined by this higher level characterization captured in the 2D space. Whereas the Z factors kind of give you more refined cell state characterizations. In particular, in the same sample that I showed, we see that it nicely aligns this green trajectory, healthy trajectory, cell maturation with healthy cells maturing to on this green trajectory, and also aligning that with the purple trajectory of AML maturation with the correct order of the cell types. And then the second axis is telling us what's most different and distinct between the two samples, and exactly aligns with the accumulation of the NPM1 mutation on the Decipher2 axis. In particular, we find this PROM1 marker marking the progenitor-like population consistently in all of our AML patients. So potentially a marker that could be used for guiding early detection of the disease. But now we can use also these axes, Decipher components, to understand what are the mechanisms that are disrupted along the leukemic initiation with NPM1 mutation. I'm going to skip this. We also compared to a wide variety of methods to try to assess how well Decipher gives a faithful joint representations by defining two metrics. We wanted to assess if these methods give us the correct ordering of cell types along the trajectories, while also, preserving the divergence and bifurcation of the trajectories and not forcing them to overlap. And so, for example, we see some methods like UMAP, they're great at preserving the order, but not as good in preserving the divergence, whereas other methods like SCVI are the opposite, and Decipher kind of has a good balance between the trade-off between these two. We also have a basis decomposition that then looks at, so the nice thing about this method is you also can get explicit reconstruction of expression directly from the model. So we can, any point in this 2D space, you can predict what is the gene expression of a cell that might be in that space. And you can basically look at now how genes change along any trajectories across clusters. And then we have a basis decomposition technique that also quantifies difference between expression along a perturbed or diseased trajectory and a healthy trajectory, basically identifying the most dominant patterns as basis functions that we learn, and then using the weights to quantify the distance between them. I think I'll skip this, but very briefly to show that we found a lot of interesting insight into disrupted regulation during AML initiation. We see, while we in the healthy sample, we see a lot of coordinated TF expression. This coordination is completely lost in the leukemia samples, and we rather see especially multiple homeobox Hox transcription factors around the time that NPM1 mutation happens. We validated these with single cell ATAC-seq as well. I want to end with saying that we started building Decipher with this question of trying to understand cancer initiation, but we found that actually the representation we get by allowing the latent factors to be correlated gives us a lot of meaningful insight into other contexts. For example, this is T cells infiltrating melanoma tumors from 33 patients undergoing PD-1 therapy. And you can see in the UMAP, they're just, they're when we annotate them, for sometimes I'm sure you've seen this CD8 T cells and CD4 T cells, they become mixed. They're not like, partitioned properly, and you don't see any association with the timing of the therapy or response. Whereas with Decipher, you get this nice alignment of CD8 T cells going from naive CD8 to effector memory and exhausted CD8 T cells, and in parallel, you have the naive CD4s going to more differentiated CD4 T cells and regulatory T cells as their own distinct cluster. Now, these axes are meaningful because we now see that they are actually associated with time of therapy, with response to anti-PD-1, and the length, the duration of the therapy given to the patient, which really tells us, can guide the administration of these therapies to be more effective. And we also get additional insight from the latent factors. This is very new. I just received this figure last night. This is new work that we just started with Bianca and led by Joy, extending the idea of Decipher to also integrate single cell ATAC-seq data. And we just see very interesting results in the context of pancreatic cancer, where you don't get the correct trajectories of acinar cells transforming into pancreatic cancer cells. From the integration, you are able to resolve this correct transformation through ADM and PANIN cells. So just summarizing, a deep generative model for the joint hierarchical representation of cells, for example, from two disparate contexts of healthy and disease. And this two-layer dimension reduction really helps with interpolating between different degrees of nonlinearity and gives you much more flexibility and therefore more interpretability. The biggest bonus is that you get a direct 2D visualization. Feel free to try it. We'd love to hear about your experience with this method. It is now online. And I want to end with thanking the many people who are actually doing all this work, many people to thank in my lab, collaborators, especially at Sloan Kettering, Sasha, and Dana Farber, Kathy's lab, funding sources, patients for their samples, and most importantly, we have open positions, especially the postdoc position joined with David's lab, where we're looking for causal inference, cancer genetics, cancer genomics, any interest at the intersection of those. Feel free to talk to us, and we'd love to chat more about it. And thank you for listening early morning.
All right. I think we have a few minutes for questions.
Great talk. So is there any time you wouldn't want to use Decipher versus using STVI or, doing Seurat UMAP?
Yes, when you have technical batch correction. So in cases, we have one data set where we have 3' and 5' chemistries. We want to align them because we know they're similar. So technical artifact, but Decipher, at least this version that doesn't do a batch correction, does not fully align them. So in that case, something like that something like STVI makes more sense.
The cell types. I'd like to get a sense of the resolution of the cell types, how many cell types and what kind, and how did you do the mapping between a cell and then cell type?
Yeah, great question. So if you have prior markers you want to use, if they're refined, it will use those, try to disentangle them. It quantifies if they're overlapping a lot in terms of the archetypes, and then it will collapse them. How the number of archetypes are chosen is based on an intrinsic dimensionality metric that we built in. So it's really a data-driven calculation of number of cell types. But we have seen in all of our tumor samples, melanoma, breast, lung, that it is able to disentangle very refined immune cell states.
Yeah, thank you. Very nice talk. One more question on Decipher. So I think you briefly mentioned the identifiability issue. So I was wondering, how did you make sure that the model will not simply ignore one of the latent representation and degenerate to a vanilla variation of autoencoder?
Yeah, great question. So we don't have theoretical guarantee that it is identifiable, but because the space of the latent space is really much smaller, so the Z space is really 10 dimensional by default. We've tried up to 30 as well, similar to SCVI. It's just because it's compressing 20,000 dimensions to 10 dimensions, it will try to make them as independent as possible, but also correlated if needed through this additional layer of B. We just see through practice so far that we get very robust representations of Z factors and also the ordering of cells along the trajectories that it infers through this minimum spanning tree. The length of trajectories in the V space has also been meaningful, again, through practice. We are thinking of ideas on how to actually try to understand what are the theoretical backings for this, but that's a good question that we're following up on.
Any questions for Elham?
So I was curious, with Starfish, you're deliberately not using a reference. If you have a reference, are you able to leverage that and then learn additional cell type archetypes on top?
Exactly. So there's three options. One is if you only want to use your reference, you have high confidence in your markers, you can just use that. The second option is you have no markers, you just want to use archetypes. The third is combining both, so adapting your marker set. And it doesn't use the level of expression of the gene markers either, so that therefore you don't need really single cell data. Even just gene names, lists of genes, even a few marker genes are sufficient.
Okay, I guess in the interest of time, we'll need to move on. So let's give Elham another round of applause. Okay, next up, we have Yichun Chen from Stanford, who will give us a talk on using ChatGPT-based models for genes and cells. That should be good.
Hey everyone, thank you so much for the opportunity to talk here, and I'm really excited to kick off our today's discussion by using a lot of interesting ideas from natural language models to apply it to single cell data. And this is joint work with my postdoc advisor, James. So to start with, the conference so far has provided us with a really clear landscape about what the current biomedical machine learning is I guess these days, where we see that we really have these really huge models also train out massive data as well. We obviously know about the massive ability of the natural language processing models, but we also learned a lot about the protein language models from yesterday's talk about their ability to take in an amino acid sequence and then really encode the underlying biology that encodes not only the function, but also the downstream folding properties and evolutional information that doesn't seem obvious to even a human expert. And we also have seen much more recently a lot of these foundation models that are built on single cell data. So two of the names that you might be familiar with are referenced over here, GeneFormers and SCGPT, where they leverage this massive amount of data and claim that they're able to provide a really good understanding about what these RNA sequence is going to do in the downstream biology as well. So in particular, if you look through the recent literature, some of the features are going to really emerge out of these so-called single-cell quote-unquote foundation models, and I really categorize them broadly into 3 different features. The feature number 1 is these models are often, as you would expect, large. So they often consist of a bird or transformer-like style where you pre-train a massive amount of data using multiple transformer blocks and attention layers trying to capture the co-expression patterns of genetic information. And the second feature, probably also not surprisingly, is the fact that to achieve successful representation, you really need a massive amount of data to pre-train on. So in this particular cases, both of the methods have trained on more than 30 million single cell data that are often collected across multiple studies and spans multiple organs and tissues of the human body. And the final feature, which to some is the defining feature of the so-called quote-unquote foundation model, is the fact that these models, the representation they learn can really be adapted to a wide range of different tasks. So in particular, for instance, some of the tasks that the authors have demonstrated include cell type annotation, gene functionality prediction, as well as batch integration and batch effect correction. So with this in mind, often we know that there's no free lunch in the computation land. So a natural question that arises is that in order to collect and just even process these 30 million cells is a huge amount of undertaking on its own. And we also know that trying to train these large models on massive amount of data is also very not only computationally intensive, but also involves humongous human labor just to make sure that everything is working and there's no bug in your code. So with this in mind, the motivating question of our study is really trying to understand whether we can leverage some of these off-the-shelf natural language model that's really impressive out there already and simply adapt them to gene and cell functionality prediction or for more biological tasks without having to retraining a model from scratch. And to really motivate our approach, we took a step back and think about what a gene is. So if you talk to a biologist in a Bio 101 class, they'll tell you that a gene is simply a list of nucleotides that are consisting of ATGC that can encode information of, for instance, particular regulatory elements or proteins that can be passed to its offsprings. So this is indeed the approach that a lot of the DNA language models are taking, where the point is that the input will be a certain nucleotide sequences, and the hope is that the high-dimensional representation it learns will be able to capture the downstream biology. Of course, you can take a bit more direct approach of single cell data, where often from a very abstract level, single cell RNA-seq gives you these measurements about single cell resolution data that are represented by a cell by gene matrix, where the value of the matrix entries simply represent the relative abundance of the cell at a given transcript. And this is indeed the approach some of these single cell foundation models are taking, where the point is that we can use very complex and expressive deep learning architectures to learn the co-expression level implicitly from this data. What James and I did was essentially taking a very much orthogonal approach by saying that actually we do know a lot about these genes, but not only from the implicit co-expression level, but also through the fact that there's numerous experiments being done, and there's a lot of human power going into understanding and curating what a gene means. So to give you a concrete example, here is an expert I took from GeneCards information about this particular gene that encodes protein CD24, where we see that in the first sentence, we already know that this gene encodes a protein. And later on, we also know that what kind of pathways and gene annotation is this protein involved in. So you can very much think of this as an expert annotation that really extract the more fundamental information that people have sifted through from the first or the second view of this data. Now, motivated by how rich of these information can be, our approaches is also super simple. To arrive at the GenePT gene embedding, we simply took the NCBI description of the genes. So an example is shown over here. We're then just going to pass them through your favorite large language models. So in our initial paper, we tried GPT embedding, which is the ADA text embedding, as some of you may know, that powers GPT 3.5. But often you can really pass it in through various different ones. You can choose your favorite biomedical specific embeddings. You can also just even use Llama 2. It turns out that they all achieve really good performance as long as they encode the rich information about the text. And once you pass it through the large language models, obviously you can get some sort of text embedding about the text that describe what the gene does. And then you can subsequently use that for all kinds of information that you care about the gene. For instance, we tried to cluster these embeddings. We use the embeddings to demonstrate that they encode gene-gene interaction network. They could also predict protein-protein interaction for protein encoding genes. We also demonstrate that on a subset of tasks that really encodes specific biology functions of a particular subset of the genes, using our embedding also achieves state-of-the-art performance for those specially curated genes. So just to now that we have these gene level embeddings, a natural question for the single cell data is that that's often not quite enough, right? We also want to make sure that we have a good representation on the cell level. And with this picture in mind, a very natural idea, now that we're given a matrix of cell by genes, is to simply say that we have a good representation for each individual column, because now we know that for each gene, we have this rich representation that's directly encoded by its text. It's a very natural thing to do to simply say that now to get a representation for a cell, I'm just gonna take a weighted average of the gene embeddings that we have, with the weight being the normalized abundance level for that transcript we detect in the single cell data. This will, by some algebra, amount to a simple matrix multiplication. So in here, if you have this expression data and you have the corresponding gene embeddings I just showed you from the last slide, you essentially just get a cell embedding for free by multiplying these two matrices. And we are naming this approach GenePTW embeddings, where W emphasizes the fact that we obtain these cell embeddings by taking a weighted average of the gene embeddings we obtained. And of course, now you have a representation for each gene you have. You can do all sorts of stuff that you might be interested in other canonical computational biology tasks. You can try to annotate the cell type, do batch integration, do cell clustering, do disease state prediction, so on and so forth. Of course, this is only one way to get a weighted average out of the gene embeddings we have. You can also think more about like a linguist and think about how we can directly embed single cell as language or sentences by itself. So this motivates our second approach, where now, again, coming back to the single cell expression matrix, if you just zoom at one row at a time, what we're going to do is to simply say that the cell can actually be represented uniquely by a sentence, where the sentence is essentially the concatenated list of gene names, where the order is determined by the normalized expression level from the highest to the lowest. So in particular, we're trying to use this string representation for each cell, because now you can pass it again through your favorite large language models. But also in addition, we think that this ranked approach really encodes a lot of information, because where in the sentence will a gene approach is essentially what we're trying to do in for instance, in using the canonical differential expression test kind of technique when looking at the raw matrix data as well. So now if we pass them through your large language model, I don't have to really belabor the details. You again get the embedding, and these embeddings, we're calling them GenePTS, where S emphasizes the fact that they're encoded from this sentence representation of a single cell data. Now, with all these approaches I've explained to you before, it's only natural to understand how well they do. So the first set of results we're going to show is that through a quick sanity check, these gene embeddings actually do encode a lot of biological information. So in particular, over here, what I'm doing is that I took the gene embeddings for more than 30,000 genes that we have in NCBI summary, and then I did a UMAP visualization of the embeddings colored by gene functionality. So over here in this plot, each dot is going to be a gene, and its corresponding color is going to be essentially what gene functionality course class they're in. And you can see that different color dots are clustered and pretty separated. So that's pretty good news that our embedding are actually encoding some of these information. And we also did a sanity check with a very simple classification task, where we took the top 15 abundant classes of these genes and just put a logistic regression on top of the gene embeddings and trying to classify the genes. It turns out it outperforms the previous baseline that essentially uses a dense co-expression data by more than 10% in terms of the accuracy. So far, so good. And then we took this approach and went on to understand whether we can predict gene-gene interaction network over here. So in particular, this data set we took, they transform or cast the gene-gene interaction problem as essentially a binary link prediction, where the goal is for a given pair of genes to predict whether they have a shared common gene annotation ontology that's not extremely common among the two. And the negative examples are just taken from the ones that don't really share a Go annotations. And in particular, just by doing a very simple featureization. So in this case, what we did is we simply took the gene embeddings through our approach for two different genes and then add them up to preserve the permutation invariance about the pair, and then pass it through a very simple random forest classifier, and we get state-of-the-art performance compared to previous dense embedding as well. In particular, one other thing I want to call out is we have a diagonal line of red. I'm trained as a statistician, so I'm always afraid that when I'm having high dimensional embeddings, I'm going to overfit. So we also did a negative control, where essentially what I did with the red line here is that I sampled the same dimension of Gaussian random noise for each gene, just to make sure that through this approach, we're not overfitting to the fact that we're simply having more dimensional data coming out of our large language model embeddings. With this in mind, I will quickly go through the next slide. The message is also pretty clear. In addition to gene-gene network that was derived from Go annotations only, we also tested the predictions on protein-protein interaction data and demonstrated that on various data we've looked at, this language model consistently performs competitively, if not better, than previously densely derived embeddings in this case. Now I have a giant table over here, but don't worry, before you panic about how many numbers you need to read, I'm going to tell you a set of numbers you need to pay attention to. So over here, what we see is that I have some classification tasks represented in 4 of the different columns, and the number reported are the cross-validated AUC through 5 different folds. What we have on the top is a gene former, which is a fine-tuned transformer model that particularly really looks at the single cell data and then fine-tune on these 4 different prediction tasks. What we have on the bottom are different gene-PT variants. The top 2 are going to be encoded through GPT 3.5, and the bottom 2 are going to be encoded through Llama 7 billion. And we also have either a logistic regression and random forest, but they perform, as you can see, very robustly, very similar to each other. So the point of this slide is really to tell us that through these 5 numbers, GPT gene embedding achieves really competitive results compared to some of these more fine-tuned and even very, very specifically designed models on these biologically relevant tasks. I also have a second set of numbers just as a sanity check. So over here, what I have as before is also that I did random embeddings of the same size, just to make sure that on these tasks, which are biologically relevant, but also much smaller in scale, we're not overfitting just because we happen to have a larger dimension. So through an ablation study, by removing the entire context about what we're embedding, we see that really by using these gene information, we're able to encode a lot of the biology that people do care about as represented in these prior work. Now, moving on from genes to cells, we also have some data, which I'm also only displaying partially, that compares the performance of GPT cell embeddings with some of the more state-of-the-art models like SCGPT and GeneFormer. And we can see that while we don't always do the best, so this is only showing one particular data set, you can find the full analysis in our paper online, we still do really competitively, especially the sentence embedding. And one other thing that's pretty interesting, and it's an active ongoing source of work, is trying to understand whether we can combine these 2 sources of information together. So if you recall in the second before, some of these larger single cell foundation models that's out there really takes advantage of, we have a ton of cells and we're learning the implicit co-expression data between the genes, but we're taking a more direct approach, which can almost be assumed as orthogonal, because we're only looking at the prior work that describe what these genes does without really looking at the observed data that we have. So a way of combining them together would be really interesting direction of future work. And what we're showing here is that if we ensemble the predictions we get from these different sources, we do seem to get better performance, at least on some of the data sets that we've looked at for cell type annotation tasks. Now going on, it would be remiss if I don't show that these models can also do batch integration and batch effect correction. So over here, we can see that I have this data set taken from heart aorta patients. On the top, I'm showing the original single cell RNA sequencing data. The left column is querying the data with the phenotype that we observe for the patient. And on the right column, we're showing the cell type that's being annotated from the original data. And we can see that there is strong batch effect present in the original single cell RNA seq because different cell types seem to be segregated from each other, even though they really are annotated to be the same. And on the bottom, we see that by transferring these information into a sentence of strings ranked by their expression value, and then embedded them through any large language models, being GPT 3.5 in particular here, we're able to remove batch effects, but also retain a lot of the biology here, because we do see that the cell types in the bottom are much more like clustered compared to the top. Now, in conclusion, what have I told you today? I've told you that the large language model embedding in our study provides a simple but very effective way to predict the things we might care about cell and gene data, especially in the single cell landscape. There's ongoing extensions, as I mentioned before, to make this approach more context aware, but also trying to understand how it could be integrated into these large scale atlas-like data to enable fast and efficient retrieval tasks. And in conclusion, I also want to say that even though we've only tried this for single cell data so far, we do think that rich textual descriptions in general can provide a very useful and very high signal modality, and we're really excited to see whether this idea can be replicated in other computational biology field as well. With that, thank you so much for attention, and I'm happy to take any questions you might have.
Thank you so much. This was a great talk. So you showed that the model does a great job in removing the batch effects, but you're not using any explicit encoding of batches or doing anything. It just does that as you train it, right?
Right, that's correct. We didn't give it any batch information.
So why do you think that is? So let's say you have the same gene, and let's say these embeddings are multiplied or somehow normalized by the count that you have. There's no information of batch. Wouldn't you expect to still see batch information there? What happens there that removes these batch effects?
Yeah, it's a really good question. It's a question that we've been asking ourselves through and throughout again. I think our current hypothesis is that a lot of the batch effects can be removed by simply considering the rank of the genes rather than the absolute abundance even after normalization of a gene. And that's like why we think it's able to detect that. So indeed, if you actually transform your single cell expression data by total rank normalization, so instead of looking at the values, you compute the rank directly, you get very similar performance to what we have, but we still have an edge in terms of batch effect correction. And I think that's partly due to the fact that you do pay different attention to the first and top bottom. So if you think about a rank perturbation, right? If you perturb the rank of the genes at the very bottom and the very end and the very top of the gene expressions, like they might have the same effect, but that's likely not the case in a large language model. But that's a great question.
Thank you.
We have time for one more question.
Thanks, great talk. I'm curious about the issue of data leakage when you're evaluating your model. Like, for example, you showed one of the motivating examples was predicting gene-gene interactions based on which the edges are based on Go terms, but Go terms were part of those NCBI text summaries that went into the model. So you basically told it the text label when you generated the embeddings.
Yeah, that's a great question. Yes, just to clarify, some of the protein functionality classes, we do expect them to do very well. So I think of them more as a sanity check to make sure that we get useful information rather than trying to prove that this is something that biologists are going to use, because, you know, like we already have these annotation, right? Like in practice, really, you're just going to look it up at the NCBI website rather than using a prediction model. But the gene-gene interaction is a great question as well. We started with this data set trying to understand whether any information is encoded, because in the NCBI summary, it's unclear whether all the Go annotations are consistently provided. So I think it gives us some comfort that we're able to predict something that's not always there. But this is indeed the motivating factor that drive us to test it on more like, I guess, protein-protein interaction data set that doesn't simply use the Go annotations. And I'm happy to take any questions offline. Just feel free to find me afterwards.
Let's thank Yi Chun again. Okay, so next up we have Rebecca Boyarsky from MIT, who will tell us more about single-cell RNA-seq foundation models. Rebecca is joining us virtually, so we're going to play a recording of her talk, and then she'll take a live, she'll do a live Q&A session.
These models give inspiration to discuss the foundation models of other areas of learning, like mapping and processing and machine learning. And the big idea is that they train transformers to learn rich gene and cell patterns using vast amounts of unlabeled single-cell RNA data, and then fine-tune models that cell and downstream functions. These models have the potential to help us discover rare cell types, learn gene regulatory networks through the transformers learn extinction rates, overcome issues like fast effects, and excel at downstream supervised tasks like cell type annotation and perturbation response prediction, even when limited training data is available. In the work I'll present here, we probe the capabilities of this model to deliver on some of these promises. Before I move into our analysis, I want to give a quick high-level overview of how transformer models are used in small single-cell structure data. So each cell is represented in the model as collected shown here. Each box represents a gene, and there are two pieces of information that the model know about each gene. First, which gene is it, represents a gene and called genetic entity that is considered. And second, how highly expressed is that gene, represented in blue. And the expression of each gene is big as a key function. These gene inputs are fed into transformer layers, which are the major building blocks of each model. The transformer layers apply attention in order to learn contextualized gene things, in which each gene representation is influenced by cellular context. So context refers to the expression of all the other genes in that same cell. So how are these transformer layers trained? These have varied between methods, but some sort of mass pre-training is deployed. During pre-training, the expression of some important genes is masked, meaning hidden from the model. And the model trains with the context of all the other observed genes in order to predict the expression of the mask. In theory, this should force the model to develop a good understanding of gene-related inputs in order to be able to excel at the task. And the weights learned during pre-training to provide good initialization of the model on all the downstream optimization concerns. Each cell can then be represented either by aggregating contextualized gene environments or through training and Frequency is often followed by fine-tuning in which a small amount of weighted data is further training models to excel at particular values. This is shown here, where the learned contextualized gene patterns are used as input to a smaller neural network, which is trained to predict some cell level weights, such as cell type or response perturbation. The full network, including the transformer, can be back propagated through reinforcement learning, updating all weights in order to improve the particular prediction. Fine-tuning allow us to leverage massive amounts of unlimited data to help improve performance around certain tasks, but these models come at a cost. In particular, taking SCGPT as an example and comparing it to a simple model of logistic regression for the fine-tuning path of cell type annotation of 3,000 instructions, we see that SCGPT has over 51 million changeable parameters, while logistic regression has just 3,000. SCGPT requires training on a GPU, which can be a barrier to entry to many researchers, and for the particular data set profile here, training took over 300 times longer and took 17 gigabytes of GPU memory. And it's whispered into the 3,000, but it would be reasonable to want to use all 20,000 participating in the model, in which case these numbers will also be changed. Furthermore, this slide describes fine-tuning, but frequently even more expensive technologies regarding. And there are also, of course, environmental costs associated with the energy consumption needed to train these models at a large scale. Because of these costs, it's really important that we understand where these models excel, what they should be used for, and what they're not used for. Here in this work, we take a deep dive into two such foundation models, SCBERT and SCGPT. Because of time constraints, I'm not going to explain the differences between these two models, so we can refer to the original publications or our papers for more details. In particular, we investigated the benefit of the pre-trained and fine-tuned paradigms within the law of data with a focus on the fine-tuning path of cell type annotation. We noticed that the papers that introduced these models didn't compare their cell type annotation results to one of the most simple baselines, regularized logistic regression. When we compare against this baseline, we can find that more often than not, logistic regression is comparable or outperforms their local foundation models. Still, one promise of foundation models is that the representations learned during pre-training might allow you to perform better at initial training, or training with only a little bit of information. With this in mind, we also tested whether the foundation models would excel as we reduce the amount of training data available. So we still found that logistic regression is often superior in the detailed testing. Let's take a look at these results. There are 4 main data sets used for the task of cell type annotation from the SCBERT and SCGPT papers. SCBERT reported results for a 68,000 cell PGMC data set. One size. SCGPT reported results on 3 different data sets: multiple sclerosis, which contains neuronal cell types from healthy donors from patients with MS, a human pancreas data set, and a data set of tumor-infiltrating myeloid cells from 9 different tumors. The size of the train and assessed for each data set are shown here. And it's worth noting that the multiple sclerosis data set is split in such a way that it represents a case of data set 6, where the task distribution is significantly different than the train distribution, because all the healthy donors are put into the train set, and the MS patients are put in the test set. So this represents a particularly challenging case for doing prediction. Okay, here we have our first set of results. SCBERT compared to logistic regression for cell type annotation with PGMC data. It's hard to see, but there are multiple dots on the plot for each model, representing runs over different. So models trained from scratch, or fine-tuned from scratch with different randomization settings. We report the macro echelon score, which is the harmonic mean of precision in recall, and which is a good metric for classification when you have unbalanced classes, which is the case in all of the data sets here. As you can see in the plot, and perhaps more clearly in the table, logistic regression outperforms SCBERT for annotating flat cell types. Next, we ran a logistic regression baseline for all the data sets evaluated in the SCGPT paper. Starting with the multiple sclerosis data, we found that SCGPT did in fact outperform the logistic regression baseline. One possible explanation for this is that multiple sclerosis data is the data set that suffers from the PGMC. The pre-trained model might actually be uniquely suited to handle this kind of situation, since it's seen many different clinical contexts during pre-training. While the district regression was only trained on a single clinical context that is no longer heavily tested. In other words, it's possible that the pre-training server becomes a regulation that protects the foundation model from overfitting to the training distribution that it's trying to use and helps it to generalize. So the pre-trained data, the latest regression is compared with SCPC. We find that the metrics are fairly well matched with the model. And finally, for the myeloid data, the district regression is slightly outdated. For our fever experiment, we subsampled each training data set to the 10, 25, 50, or 75% of the result size, as shown on the x-axis of these plots. For the most part, we found that the model was outperformed with the full training data, also outperformed in the p-top setting. For the pre-trained data, where the district regression and CBCC is very closely tied with the full data, we found that actually the district regression was significantly better than CBCC in the p-top setting. So, unlike our hypothesis that the pre-trained model would have a unique advantage for T-cell learning, we found that with district regression, with two parameters, actually fared better in training data. While the district regression is competitive for the fine-tuning of cell segmentation, foundation models still have other key benefits over the district regression. One being that they learn gene and cell embedding, which might capture meaningful biology and be useful for others. With this in mind, we ran some efficient studies to better understand the right implication of capabilities of the foundation model. First, we tested the effect of removing pre-training altogether to directly evaluate how much pre-training helps with the fine-tuning past of cell segmentation. Here's the visual of the experiment. You'll recall that I explained in the foundation model that in these models, the ratio of transformer layers are initialized using. Instead, in this experiment, we initialize the transformer to have the red. And we then proceeded directly to training and over predict cell types in the different data sets. For SEBERT, we found that there was no difference in performance for imitating the previous two cell types with or without pre-training. For SCBCC, we found that the pre-training did benefit the model. We need some further follow-up analysis to understand what accounts for the differences in efficacy of pre-training between SEBERT and SEBERT. Finally, we present a pulmonary trial showing that good pre-training performance can be different and does not always mean that meaningful data representation is different. In this experiment, I'm going to give a little more detail about the way that input data is processed in the SEBERT model. As we said earlier, each gene is input as a sum of a gene identity embedding, representing which gene is which, and an expression embedding, representing how highly each gene is. The expression embedding actually represents in the expression data, and so it maps to a category. In the case of SEBERT, they bin the data according to the following 6 bits. And note that these bins are pretty close. All gene expression is categorized into one of 6 bins, where UMI stands for unique molecular identifiers, which is a measure of chemotherapy. During pre-training, the transformer layers are used to learn nuanced representations of each gene given the context of its cell state. Each input gene is unique, and that is the unique combination of the gene identity embedding and the expression embedding, and it ends up being represented by a unique contextualized gene embedding after passing through the transformer. Each of the masked genes, shown here in black at the bottom, end up having different contextualized representations, since the gene identity embeddings differ for different masks. This allows the model to make a unique prediction for the value of each masked gene, as shown here at the very top of this map. In our ability experiment, we removed the gene identity embedding completely from the input gene, so that only expression embeddings remain. In this setting, the model has absolutely no notion of which gene is which. And recall that transformers are permutation inhibitors, so the model can't even use traditional information to deduce which gene is which. The only information that the model has about each gene is this expression bit itself, one of the six expression bits. And therefore, every gene with the same base expression will have the same chain that is output by the transformer. Similarly, every mask gene will necessarily have the same representation output by the transformer. And the model will be forced to make the same prediction for every mask gene in a cell during the It seems like this should be an impossible situation for the model to do well at pre-training, since it doesn't even know which genes make the prediction. But what we found is that the model actually has pretty much, while the regular SQL model achieves 78% accuracy on the mask, this severely limited model will achieve 72% accuracy. So we got ourselves wondering, how is it possible, given that the model does not know which gene is which, that it still does fairly well at predicting what the mask is. And digging in, we found that in this setting, the model learns to always predict that the mask value is equal to the most common bin for that cell. Since there are only 6 bins, and some bins are more common than others, this leads to a pretty high accuracy of 70%. This information study is meant to serve as a cautionary test, but if the pre-training task is too easy, the model can learn simple heuristics to do well for pre-training without actually learning the rich gene representations that we're after. This underscores the importance of designing challenging pre-training tasks that encourage the model to learn representations that will be useful and informative in the ways that we like. And it also highlights the importance of careful introspections into your model to make sure that it's actually learning what you need. If you use this in mind, I want to wrap up with a few key takeaways about it. First, it's important to compare deep models to simple baselines to understand the which tasks they're truly using. Second, since simple baselines are often comparable or better than foundation models with cell-type annotations, we need to move beyond cell-type annotations to showcase the value of foundation models, which SGBTP, Beamformer, and other papers are already doing. Foundation models might really favor this when it comes to more challenging fine-tuning tasks, which simple baselines can't do well at, zero-shot capabilities, or investigating gene interactions and gene regulatory networks through their learning. We also show here that strong pre-training or fine-tuning performance does not necessarily mean that this favors representation of the learning, and this calls for careful introspection into models as well as for careful crafting pre-training objectives. Actually, we didn't show the results here, but in our pre-training we show how a hyperparameter setting and random foods can have a really big effect on model performance. And therefore, detailed and transparent reporting of all of these settings is critical in order for the community to get a reproducible opportunity. I'd like to finish the following funding sources and contribute all pages for feedback. All of our analysis code is available on GitHub, and we will check it out.
Rebecca, are you there?
Yes, hi.
Okay, so all right, so we have time for a few questions for Rebecca.
A great talk. I think it's really great work and important to benchmark these models that sometimes I think assumptions are made about. So with your kind of caveats around the cell type annotation benchmark, what do you think are other good benchmark tasks to evaluate these foundation models?
Yeah, I think that's really one of the salient questions. So first of all, I think it's possible that there are cell type annotation tasks that are a lot more challenging than the ones that have been presented so far.
Rebecca, can you hear us?
Yes, I started answering. Can you hear me? Hello.
Wait, is that right? Or it should be that?
So, okay, can you hear us now?
Yes, can you hear me?
No, sorry.
Can you hear me?
You're gonna have to repeat the question.
We
Yeah.
can hear you. Did you hear the question?
I got the question. Yes, yeah. Okay, so you can hear me now. Great. So yeah, I think that's really one of the salient questions. So first of all, I think there's cell type annotation tasks that are probably harder than the ones that were presented in these initial papers. So for example, cells from like very different tissue contexts might be much more challenging to annotate, like really kind of out of distribution cell type annotation. That's one thing. The other thing is, and I think this is coming up in the next talk, but looking at zero-shot capabilities of these models. So if we can learn sort of universal embeddings that really give us fine-grained information about cell types, the ability to discover rare cell types, things like that, that would be really useful. And then also, I think introspecting on what the model's learning in its attention weights, in terms of like this could really kind of be a breakthrough in ways to discover gene regulatory networks. That would be amazing from a biological discovery perspective. I do think that I've seen other people say this as well, that these models right now are a bit of a hammer without a nail in terms of, I think it's hard. I've spent some time trying to come up with challenging downstream tasks, and GeneFormer and STGPT have a few other downstream tasks that it's worth looking at, but I do think it's hard to come up with downstream tasks that are really hard and that these models are good at currently. So I think that's one of the open directions for this field.
Hi, that was a great talk. Thank you. To what extent do you think that transformer layers are making a difference in this type of model?
Yeah, so that was what we tried to get at with the pre-training ablation study. And it's a little hard to disentangle since we learned in the logistic regression part of the talk that the cell type annotation tasks done here are actually pretty easy. So for example, in SCBERT, we took away all the transformer layers, basically. We made them just random embeddings. And we found that the model still does well, but that doesn't really tell us that the transformer layers aren't good for anything, since the task is very easy, that it just might not need those layers for this task. I do think that with harder tasks than running these ablation studies of seeing with and without the transformer layers, how good are the models, that will really get at, are the transformer layers contributing to the good performance of these models? And then also the transformer layers are where kind of all the meat of the attention mechanism is, and where potentially we could really be learning gene-gene interaction. So I just think seeing a focus in the future on what is being learned in the transformer layers, like really deep introspection into the representations learned, will be very informative and probably require some creativity in terms of different ways of looking at what's being learned there.
I think that's all the time we have for questions. Yeah, so let's give Rebecca one more round of applause.
Oh, I just want to say one more thing for people on the live stream. I know the audio didn't come through, but I'm sure I and the organizers of this conference will find a way to share the video afterwards. So sorry about that, and I hope they'll be able to watch the talk later.
Yeah, we will be posting another full version of the recording online after. Okay, so next up we have Kasia Kędzierska from Oxford, who will talk to us about zero-shot foundation models in single-cell biology. She will also be remote and also do a live Q&A session afterwards. Yeah. Advanced.
Yeah, maybe.
Oh, sure, sound.
Yeah, I think that's why.
You're brave enough. Probably shouldn't do this. You think it works? Yeah. Okay. Okay. Yep. We'll get started.
Hello, my name is Kasia Kędzierska, and today I'll be talking about assessing the limits of zero-shot foundation models in single-cell biology. This work was carried out during my internship at Microsoft Research in England under the mentorship of Lauren Crawford, Ava Amini, and Alex Liu. When we dissect a tissue, we get a set of different cells, and before the advent of single-cell technologies, we only be able to get a readout across average of those cells. But nowadays, we can get a more detailed picture. For example, we can run a single-cell RNA-seq experiment and get an expression profile of each of the cells separately. Let's look at this matrix that is an output of the single-cell RNA-seq. We have cells in rows and genes in columns, and each position of that matrix will tell us about the level of expression of a given gene in a given cell. However, this matrix is quite an idealized version of a real-life one. In real life, the data is much noisier and sparser. And the challenge lies in uncovering the patterns in the data. We could imagine having a model that would take that matrix and create an embedding space where we could project the data. And what we would see, for example, those 4 clusters. And we can get excited about potential exploring the differences between those clusters and learning about the biological differences. However, what usually happens is that batch effects confound the data. And if we would not know about the batch effect in this data, we would focus on the technical differences rather than the biological ones. And the goal is to learn about the biological, about cells and their identities. So what we want a model that would create an embedding space, a different embedding space, such that would reflect the biological differences in our data. And recently, due to popularization and success of systems such as ChatGPT, a new type of model became popular within the single-cell biology community. The foundation models claim to learn universal patterns from observing vast amounts of data during pre-training, tens of millions of cells spanning different tissues, tissue types, organs, and cell types. Those models claim to serve as a starting point for various analyses, such as labeling of cell types, gene prediction, gene expression prediction, and perturbation analysis, and so on. Those models include SC-BERT, GeneFormer, SCGPT, SC Foundation, and many more. They are quite popular within the field because of their potential automatization of tasks such as cell type labeling, comparing different data sets, and so on. However, because those models are quite new, there does not exist an established evaluation setup. And most of the work so far has focused on fine-tuning. What does fine-tuning mean? It means that when we take the model and either retrain all of it or parts of it on a smaller data set. Whereas where we take zero-shot, we take the model as is. Evaluating the fine-tuned model also comes with potential setbacks. First of all, we need a set of clear labels, which does not always come easily in a biological exploratory work. And secondly, it requires a complicated setup or extensive GPU resources, whereas zero-shot is accessible. Additionally, if we assume that the models learn the biological patterns during their pre-training, those should be accessible in a zero-shot setting. And that's why we decided to evaluate those models in zero-shot scenarios. We took 2 models. We evaluated 2 models, GeneFormer and SCGPT. They both draw inspiration from the foundation models in natural language processing, but they have some differences. For example, they differ in how they take the input. GeneFormer takes the ranked list that is created based on an expression for each cell, and SCGPT bins the expression. The binned expression in SCGPT allows comparison across the cells directly. When they embed the values, GeneFormer only embeds the gene IDs, and SCGPT embeds the gene IDs and the expression value directly, and then sums those embeddings in the embedding space. When it comes to architecture, SCGPT is bigger, and when it comes to training objective, GeneFormer uses masked language modeling when it predicts the gene at a given position, and SCGPT predicts the expression of masked genes from unmasked genes and cell embedding in one pre-training objective, and the other part of the pre-training objective is the prediction of the gene expression from cell embedding. And when it comes to the cell embedding, in GeneFormer, that's average over gene embeddings in a cell, and SCGPT learns the cell embedding directly as a first token. In our evaluation setup, we looked at 5 data sets. We looked at the pancreas that spans one tissue type and several batches. We also looked at PBMCs that have 1 or 2 technical batches. And then we looked at more complex data sets such as cross-tissue immune and tabula sapiens, which span multiple tissues and multiple technical batches. We also evaluated the models on 3 tasks: cell type clustering, when we asked the question how well the cell types are separating within the cell embedding space. We also asked how well the batches are integrated within the same cell embedding space. And we investigated the pre-training objective. And now, moving on to the results. First, I'm going to talk about how well the cell embedding space separates the cell types. And for that, I will use 2 scores, average silhouette width and average bias scores. Silhouette score is going to tell us about how distinct and how separate the cell types clusters are in the cell embedding space. And average bio is a composite score that takes silhouette score and 2 other scores, and also talks about how well the clustering can be performed within that space and how well those clusters align with the cell type labels. For both scores, the higher the value, the better the performance. And as I said, we will evaluate them on 5 different data sets. And to give us some perspective, we will evaluate and compare GeneFormer and SCGPT human performance to a simple heuristic method of choosing highly variable genes, and also to using SCVI, a variational autoencoder method that is commonly used in single-cell RNA-seq data analysis. Then we calculated the median across all the data sets, and what we see is that SCVI outperforms foundation models in both scores, in silhouette score and in the average bias score. Additionally, what we see is that highly variable genes outperform the foundation models in the average bio composite score. That is the same for the pancreas, which largely follows the median. However, when we look at the PBMC data sets, that is a little bit more complicated. And let's focus on the PBMC scores for SCVI and SCGPT human. So what we see is that when it comes to the silhouette score, in both data sets, the SCGPT outperforms the SCVI. However, when we look at the average bio score, we see that SCGPT human outperforms SCVI in one of the data sets, but underperforms SCVI in the other data set. And also what we can notice is the spread of the values. SCVI is quite concise within the values, and SCGPT has quite big spread of those values for PBMC datasets, suggesting that it's somewhat unreliable for the dataset coming from the same tissue but different data sources. The more complex datasets also largely follow the medians, and generally what we can see here is that SCVI and highly variable genes outperform foundation models in cell type separating. Now let's talk about batch integration. Here, we're going to focus on the pancreas dataset, and as I told you, it spans one tissue type and several different batches. In a perfect batch integration scenario, we would see clusters separating based on cell types and lack of organization due to the batch type. And when we look at the highly variable gene, we see an improvement over the space generated by all genes. We see some clusters based on the cell type and some mixing of the batches. However, when we look at the SCVI, we see a very nice pronounced clusters based on the cell type. And however, we still see some organization with respect to batch within the clusters. However, when we look at the GeneFormer, we see that the primary driver of the organization in our data is actually batch type. And then there is a lot of mixing within the cell type. In SCGPT, we see clusters based on batches, but then some additional organization based on cell types. Overall, we see that foundation models did not perform well at batch integration. However, this is just a UMAP projection. This is just evaluating this and giving us intuition. But to formally evaluate the performance of those models, we looked at the batch integration score. Batch integration score is calculated on the embeddings directly from the models or the methods. Batch integration score is a modified silhouette score where the higher value would signify a better mixing of the batches. And we evaluated the models on 4 data sets, Pancreas, PBMC, Immune, and Tabula Sapiens. And again, we calculated the median across all the data sets with the dashed line. And first thing we see is that highly variable genes outperform other models, especially the foundation models. And second thing that we see is that, for example, in the case of the gene former, there's unreliability of the score on different data sets. There is a high difference between, there is a big difference between the low score and the highest score within the 4 data sets, meaning that we can't really be sure that if it performed well for one data set, that it will also perform well in another. Overall, highly variable gene selection outperforms foundation, more complex models such as foundation models. So we saw that the foundation models that we evaluated underperformed in our previous evaluation metrics, and we wanted to see what happens in the pre-training. So first, we're going to look at SCGPT, and as I mentioned earlier, SCGPT has 2 training objectives: gene expression prediction, when the masked gene expression is predicted from unmasked genes, and cell embedding. And we're going to look at the input expression bin and predicted expression bin. And what we see that in this first pre-training objective, SCGPT actually outputs an estimate similar for all of the input expression bins. That improves when we take the gene expression prediction from the cell embedding, GPC, with the more variability across the predictions. However, to get a more formal evaluation, we looked at the mean squared error, MSE, and to give us a baseline, we calculated the mean input expression bin for each gene across all cells and used that as a reference. So here we plotted in the dashed line, we plotted the median across all the data sets for the mean bin prediction. And if the model will perform better, those values are going to be below the line. And if the model is going to perform worse, the values are going to be above the line. And what we see, as expected, SCGPT random, which is a random initialization of the model, performs worse, with GBC performs worse than GEP. However, when we look at the SCGPT kidney and SCGPT blood, which are a variation of SCGPT pre-trained on kidney dataset and blood datasets, we see that those values improve over a random model. And we also see that the GEPC objective outperforms GEP, which is an inverse of what we saw in SCGPT random, suggesting that the model learns the cell embedding. We also see that only GEPC objective for blood and human outperforms mean, and only in several datasets, not all. When thinking about gene former, because of the nature of the output of the model, we will be comparing the input expression rankings with the output, with the predicted expression rankings. And what we see is that we have a lot of points on the diagonal, suggesting that gene former performs well at predicting those values for quite a lot of genes. But we also see a lot of points on the bottom of the plot and on the left, which suggests that gene former also tends to predict genes that were not present in the input ranking, or not predicting the genes that were present in the ranking. To formally evaluate it, we will be looking at the Pearson's correlation between the input and the predicted expression. And to give us a baseline, we calculated the average ranking. So for each gene, we calculated an average position in the rankings present in the input. And then in the dashed line, we have the median for all those data sets in the average ranking. And again, and here, when we're going to see the values above the line, that means that the model performs better than the average ranking. And if the values are going to be below the line, that means that the model performs worse. And what we see is that GeneFormer outperforms the average ranking in PBMCs, but underperforms in other data sets. In summary, the cell embeddings generated by the foundation models struggle with cell type separation in zero-shot scenarios. They are susceptible to batch effects, and that the current approach to pre-training may offer limited advantages for model performance. I invite you to read more about our research in our preprint, Assessing the Limits of Zero-Shot Foundation Models in Single-Cell Biology, available on bioRxiv. And I would encourage you to see the version 2, where we updated quite a bit of things. And I would like to take this moment to thank Alex, Ava, and Lauren. I loved working with you, and I would like to thank the whole of the MSR New England Lab and the interns cohort of 2023. And to you for listening, thank you very much. And I'm happy to take your questions. I'm here. I don't know if you can hear me.
Yep. Can you hear us, Kasia?
Yes, I can.
Great. Questions?
That was a really great talk. I guess your talk and the previous talk highlighted how the current pre-training tasks might be too trivial and not super informative. So I was wondering if you had any thoughts about, what might be more appropriate tasks, more challenging and more informative.
Yeah. So generally, I think that first of all, we should actually evaluate those models on the tasks that we presented and what Rebecca presented as well. And it doesn't mean that the models need to perform fantastically, but we need to understand what they actually learn in the pre-training, right? And we need to find out whether they can perform at those tasks and what they cannot do, and why is that, and what maybe they are missing at the pre-training stage, right? I like this thing that Rebecca mentioned, that some people are saying that the foundation models are actually hammers without the nail. And I think that the onus is kind of on the creators of those models to tell us what they actually are going to be good at. I'm not saying that they are not going to be good at those tasks. It's hard to imagine what they can do in the future. And yeah, I think that so there are a couple of other foundation models that I would like to evaluate as well, like universal cell embeddings that have been published a couple of days ago. And I think this one has a good chance of performing well in the tasks that we have presented. So I still think I would argue that the tasks that we have selected are still a good evaluation to get a sense of how well those models perform.
One more question.
Hey, Kasia, great talk. I had a quick question about the pre-training steps in terms of like training on sparse data. Do you think that maybe using like pseudo bulk and like using that as a data augmentation step might help learn better embeddings, especially with like masked reconstruction?
Yeah, fantastic question. I actually think that additional work to be done with the data will improve those models further. Because I think in biology, it's a little bit harder to use the same concept that people use in large language models, when you just train on a lot of data and then come up with important and interesting patterns. But I think in biology, you need to do some kind of data curation or some kind of data pre-processing and make sure that the data fits better in the task that you the models or the tasks you want to use on it. So I think yes, maybe pseudo bulking would improve. I think that improving the quality of the data and making sure that the data is processed in the same way to eliminate some of the potential drawbacks there would be a good first step to see whether that improves, for sure.
Okay, great. Let's thank Kasia again for a great talk.
Thank you very much.
Okay, so for the last talk before our break, we're going to hear from Eli Bogart, from lead scientist from Deep Genomics. Deep Genomics has actually sponsored MLCB for a number of years now, and we're thankful for their continued support.
All right, it's a pleasure to be here representing Deep Genomics today. I know we're coming into the coffee break, so I'll be brief. I want to take the next 5 minutes to give an introduction very briefly to the company and then sort of an advertisement for some of the machine learning work that we've recently been doing that I think will be of interest to this community. So as a quick introduction, we are a drug discovery startup. Our mission is to revolutionize drug development by applying artificial intelligence methods to RNA biology. We have a fantastic interdisciplinary research team that combines strengths in computational science, especially machine learning, with experimental biology. We believe that our approach is applicable to a wide range of diseases and different therapeutic modalities. Our main focus in the past couple of years has been developing oligonucleotides designed to increase the expression of particular target genes to treat genetic diseases. So what I want to highlight here is a foundation model for RNA biology that a large group of my colleagues published a preprint on back in September. This is a model called BIG-RNA. So BIG-RNA takes in a wide one-hot encoded DNA sequence as input and fits a number of different genomic data tracks, 128 base pair resolution as output. In this audience, I don't need to spend a lot of time walking through that setup, so I'll highlight some of the distinctive features of this model. We've done a lot of work to incorporate individual whole genome sequencing data on the input side and individual RNA-seq data sets from the GTEx database on the output side. So we have data from 70 individuals across up to 51 tissues for each individual that go into the training set, leading to 2,956 individual RNA-seq output tracks. We supplement that with another 700 tracks capturing RNA binding, protein binding data, as well as microRNA binding. So we subscribe to the school of thought that says that the key thing about a foundation model is that it can be applied to a range of downstream tasks. And if you take a look at the preprint, we go through multiple tasks in a dozen different application domains, but I want to just briefly touch on two that I think will be of interest given discussions yesterday and today. So the first is that we find that the model does very well predicting the effects of variants in non-coding regions. So I've highlighted here a case study where we looked at a known pathogenic variant in the gene NAA10. This is a mutation in the 3' untranslated region of the gene, which is known to decrease expression by disrupting a polyadenylation site. And this comes out very clearly in the model's predictions. I've shown in blue the predicted RNA-seq coverage track for the wild type sequence, in red the same for the variant sequence. And you can see the decreased expression, looking at the reduced coverage in the exons, as well as this extension of a high coverage region of the 3' UTR due to the disruption of the poly-A site. As you'd hope based off of that anecdote, when we systematically evaluate the model's performance on discriminating between pathogenic and benign variants in 5' and 3' untranslated regions in the Fluenvera database, we get quite good classifiers which match or exceed the results from alternative approaches. Finally, coming back around to drug development, what we found is that the model is very good at predicting the effects of steric blocking oligonucleotides in a zero-shot framework. So for those not familiar with the approach, a steric blocking oligonucleotide basically is a way of saying, all right, I have a functional region or suspected functional region in RNA transcript. I'm going to synthesize a complementary molecule to bind to it and prevent it from interacting with its natural interaction partners and suppress its function. The model has never seen data from cells treated with oligonucleotides, but what we find is that you can very reliably get good results by taking the binding region of your oligonucleotide and just zeroing out the one-hot encoding of the sequence. So for instance, you can use these molecules to modulate splicing, for instance, skipping exons. And when we go through and pair big RNA's predictions to internal experimental data from 620 oligonucleotides skipping a range of exons, we generally see quite good correlations between the model's predictions and the exon skipping effects across those oligos at a consistent dose. We can compare this to doing a similar prediction approach using Splice AI, which is already a pretty sophisticated splicing model. And we see that in this case, going the extra mile or many miles to build a foundation model has gotten us improved performance on this task, which is really relevant to what we want to do. I'll wrap up there. Very happy that the company can support this community. Great to be here as a sponsor. If you have questions about this work or deep genomics, I'm happy to talk. I'll be around all day today. My colleague Andrew is in the back and can also take questions. We are currently hiring for a director of machine learning. If you know anyone who might be interested, that posting's on our website.
Thanks, Eli. So with that, that's the last talk before the break. So let's take a 10-minute break for coffee and then come back and we'll have another 3 talks. Yeah. Okay, sure.
Just logging the Zoom and share screen later.
Yeah, we should you should probably check that right now. Are you the next? Oh.
I'm the second.
Okay. Yeah, I would check it now because I'm not sure if you have the permissions to share.
Save Bachelor's email that I can log
Yeah,
in as
can you
a
just
fan.
join? Yeah.
It will take a second. I think I successfully logging.
Oh, okay.
I'm gonna join without the.
Can you just click on share and just see if it lets you? Okay, good. Yeah.
Okay.
Yeah, so when you basically when you bring your laptop up, we have to put these 2 in, and then I have to switch a bunch of settings on
Okay.
Zoom.
I
But it
see.
should be
Okay. So, do we need to open the audio or something else?
yeah, we'll have to do that when we connect in.
I see. So I also need to join the audio with my friends.
Yeah.
Okay.
So do you pronounce your name Gang or
Gong?
Gong? No, okay. I see.
Yeah. Thank you.
This one's not. I think that one's good. What are we? Not sure. Have batteries?
Oh yeah, there's that.
I don't know if those are fresher.
Very cool.
Works. Okay.
The question I was going to ask you is about so a bit of a massive yeah, so okay, the two things I mean, one is just no one's heard about it. The other was like tell us that tell us about this kind of like some stuff like stuff right because I think how I know everything on the website
Yeah, we did that. So we tried that. Like it's like now with more. So we did a stuff like definitely. I think something that we can it's really good at these apps, but I think it's also compounded by the fact that like people name them because they are like you know like CD8 and CD24 they have a really close embedding in any large language model because they share the structure that is the CD family. So it's a little bit harder to handle whether it's because we get them or the that's like that like smallest really pretty.
I should know that the ChatGPT what's the it's
provide you so we get you first.
But it's not word, it's like what is the art like no, it's cool, I didn't do that. What about the little bit like so we got this as like usually like you not just to like getting that really just like what and it's pretty good at it. The bad thing is it all makes it up and wants to give you an answer. So it will give you more it's really so there's no yeah like but if you're
I mean, nobody's gonna be able to do that, right? Like, they're just gonna get better. Like, someone can not entirely know them.
I'm undecided. Okay, everyone, let's get started. So the first talk of this session will be by Elio Ramani, and she's from UCLA, and she'll be speaking about replicable single cell analysis. He will be, sorry.
All right. So hello, everyone. My name is Samuel Alber. So unfortunately, Alber Ramani couldn't make it, so I'm going to be presenting on his behalf. And today, I'm going to be discussing a novel statistical framework based on random subsampling that allows for replicable single-cell analysis. So let's say we have some sort of single-cell data set measuring the gene expression of each individual cell in our set of samples. So here we have a UMAP visualization of such a data set. Here we can see the cells are clustered based on their cell type. And from here, we can do a variety of downstream tasks. And one of the more simple ones that we can do is just take two groups of cells and try to determine which genes are differentially expressed between these two groups. So one example of this is we could just take one of these clusters. We can compare them to every other cluster to find the cell type markers of that cluster. But when doing this kind of analysis, one natural question that arises is how consistent are the signals that we detect with the true biological variation in our data set. So to help quantify this, we made this graph on the left, where each point is representing a statistically significant gene. On the x-axis is the log-fold change of that gene in a large 962 sample data set that we can effectively think as a ground truth. And then on the y-axis, we're taking 5 samples from 2 separate single-cell experiments respectively. And we're comparing the log-fold change seen in those 5 samples with our ground truth. And we can see that even though we're just using 5 samples, we see very nice correlation. A different type of differential expression analysis that we can do is we can zoom in on one particular cluster. So let's say CD4 naive T cells, and we can group the cells by something like the age of the individuals that they come from. So the question is, do we still see this nice consistency between separate experiments? And the answer is no. And here we're actually increasing the number of samples from 5 to 50. And yet still we see very poor correlation for both of these separate single cell experiments. And so why is this happening? Well, on the left, we're working in a regime where the signal is particularly potent. So we speculate that it dominates any sort of inter-individual variation. And so the signal that we detect is fairly homogeneous. However, on the right, this is a regime where inter-individual variation seems to dominate the signal from age. And so the signal that we see is fairly heterogeneous. And so small data sets are not as reliable. However, this becomes problematic when you consider that if you look at all the single cell data sets published in the past 5 years and take the 90th quantile of their size, that is just 45 samples. And yet here we're demonstrating that for 50 samples, if we're examining a heterogeneous condition, we see very poor consistency. So to examine this further and why inter-individual variation matters, let's say that we have this 10 sample data set where we have 5 cases, 5 controls. Each patient in the study has different types of individual level types of variation. This could be their age, ethnicity, sex, and so on. For the purposes of this illustration, we're assuming that each patient just have 3 types of variation, which are colored in green, blue, and purple. Problems arise when there are imbalances in these types of variation. So for example, in our controls, we have 4 patients with this purple type of variation, whereas in our cases, we have just 1 patient with that type of variation. and this leads to a potential confounder in our analysis. So how might we solve this? Well, if we know what the confounders are and who's affected by it, then the simple thing to do would just be to subsample our controls and our cases in a manner that is balanced with respect to all these potential confounders. However, if we want to apply this to a real data set, this becomes impractical because often our confounders are hidden, meaning we don't know what they are or much less who they're affecting. So our solution is to repeatedly randomly subsample our data set, in this case m times, with expectation that at least some of these subsamples will be balanced with respect to all these potential confounders. In practice, we don't know what these particular subsamples will be, so we aggregate all the signals together. So for example, if we're examining something like differential expression or differential cell composition, where we're trying to see if there's some biological signal between our condition Y and our feature X, then we can take all the p-values from these m subsamples, take the maximum p-value, and note that if the maximum p-value is low, that means even in the balanced subsamples, we still see statistical evidence that our biological signal is real. Therefore, we can conclude that it is real. However, if the maximum p-value is high, then that means we cannot confidently conclude that this biological signal is real. In practice, a maximum function leads to low power. So to combat this, we use a quantile. The way this quantile is selected is that we adjust it until it achieves calibration under permutation of the case control labels. We do this separately for each feature since these types of variation might affect different features differently. Based on this distribution of p-values from these different subsamples, we can calculate a final p-value with basically the assumption that when we select a particular quantile, under the null, everything at that quantile and above should be uniformly distributed from 0 to 1 if there's no signal. And so the p-value at that specific quantile, therefore, will be the minimum of that set of uniform random variables, which can be modeled with a beta distribution. So first we apply this to differential expression analysis. We use just a simple t-test, nothing more sophisticated, since it has high sensitivity and low runtime. We compare 3 separate single cell experiments to this large 962 sample data set that we're using as a ground truth. For our heterogeneous condition, we're again using age, and then as a more positive control where we expect more methods to do well since there's a more dominant signal, we use sex and cell type markers. So if we look at, again, a data set size of 50, and we compare the ROC curves of our method to all the other methods, we can see that our method highlighted in pink for age exceeds state-of-the-art performance. And then for sex and cell type markers, it equals it. However, if we look at the false discovery rates of all these separate methods, we can see that our method in pink, which is on the far right of each one of these separate subplots, is the only method that is consistently calibrated across all 3 conditions. And to ensure that we weren't overfitting to just age as a heterogeneous condition, we also repeated this analysis with COVID-19 and lupus, where we found a similar increase in performance, if not more. So one question that arises is, we're demonstrating this for small data sets, but what happens if, as sequencing advances, data sets get larger? Does our method still become applicable? And so to test this, we took this 962 sample data set and we split it 4 ways. And then between each pair of these 4 sub data sets, we used 1 as discovery and 1 as validation, and then calculated a Matthews correlation coefficient, or MCC, which you can think of as an improved version of an F1 score. And then across all of these different pairs, we just took the average MCC. And so we repeated this analysis for all 3 conditions and saw that for all 3 conditions, our method performs the best when compared to the next 3 best benchmark methods. Okay. So then we extended this to differential cell composition. So here we're doing a similar kind of analysis, except using the sample level cell type counts rather than the single cell level gene counts. Here we're using a simple just chi-squared test as our input into our procedure. And then since the number of features here is much smaller, we can just look up each feature in the literature and make a literature curated ground truth. And then we took about 100 or so sub data sets from 4 separate single cell experiments, and then calculated their empirical FDR versus their sensitivity. And we found that our method, this robustified chi-squared, which is kind of circled in red over there, is the only method that for a data set size of greater than 10 is consistently calibrated. And furthermore, among the methods that have a similarly low FDR but aren't really calibrated, we still have a higher sensitivity. Then we repeat this analysis for COVID-19, where we saw a much stronger signal. So more methods tended to be consistently calibrated. However, amongst those methods, we can see that our method circled in red is still the only method, it's the method with the highest sensitivity in this regime. So once we apply this to differential expression and differential cell composition, the question arose of what other downstream tasks can we apply this to? And one task that we focused on was contrastive learning, where we're trying to find the parameters of our model theta that maximize the ratio of the variance of our cases with respect to the variance of our controls. And if you just kind of optimize this directly, then you might see this nice enrichment or even separation of cases versus controls, seen in the red versus the gray. However, in reality, this might be driven by some sort of hidden confounder that has an incidental correlation with our condition of interest. And so to robustify this, you can first minimize with respect to all these subsamples to ensure that the parameters that we find lead to a high variance ratio, even in the subsamples balanced with respect to all these confounders. And in this manner, we can help ensure that these hidden confounders with incidental correlations are not driving the signal that we see. So we applied this to a data set for COVID-19, where we applied it to CD4 naive T cells. Each row here is representing a separate contrastive learning method. Our method is on the top row, and we benchmark it with 3 other methods shown below it. In the leftmost column, we're just coloring the cells based on cases versus controls. We can see that for our method, we have this nice enrichment or even separation between the cells. And then while doing this analysis, we also saw that some of the genes were correlated with the directions that we found. And these led to the identification of potential subpopulations. And so the 2 columns next to it are showing the activation of those 2 genes, which are attributed to CD4 naive T cell activation. And although some of the other methods also seem to capture some structure in our data set, if we take the representations learned from each of these methods and compare them to a completely different data set, also for COVID-19, then we can see that although the other methods tend to not really replicate the structure that was seen in the training data set, our method does. In particular, the case control structure, as well as the structure of these subpopulations. We also repeated this analysis for other cell types, which I didn't include here just for simplicity. But in this manner, we're essentially allowing our method to find the variation of interest in this heterogeneous condition. So in conclusion, we developed this framework based on random subsampling that allows for more robust analysis for separate types of single cell analyses. One big kind of advantage of using this procedure is that you can just input an existing statistical model into our procedure. In fact, we use something as simple as a t-test to show that under our procedure, it exceeds state-of-the-art performance, and also something as simple as just a chi-square test, which also exceeded state-of-the-art performance for differential cell composition. And then, as you just saw on the previous slide, if you apply this same type of idea to contrastive learning, you can robustify it so that it's more replicable across separate experiments. And then in that manner, you can isolate the variation of interest that you're interested in. So in conclusion, I would like to acknowledge the rest of my team, in particular Professor Rahmani, as well as Professor Nir Yosef, who mentored me throughout the course of this project. And I'd like to thank the audience for taking the time to listen to my presentation. Thank you.
All right, questions for Sam?
Hey, thanks for a cool talk. So you've got this really nice method to account for sort of finite sample effects in a much better way, but you're comparing it to methods like DSeq and Luma, which really get their power from pooling information across features. So is there a good way to combine both of those good things?
Yeah, I think, is the microphone on, by the way? I think I can't. Yeah, actually, yeah. Can you repeat the first half of that question?
Yeah. So I was saying that you have this cool method to account for finite sample effects in a better way, but the comparisons you're making are to methods like DSeq and Lemma, where the power is really coming from pooling information across features. So can you do both of those together at the same time?
Yeah, I guess we haven't experimented with that too much. I mean, you could apply a method like that within the subsampling framework. And I think we did try that, although we found that due to power reasons, it didn't perform very well. So that's like one of the reasons why we use something like a t-test, since it's overly sensitive. But yeah, it seems like pooling cost features for now doesn't seem to kind of lead to increase in performance. But this is still, unpublished, so that might change in the future.
Thanks, great talk. How does this compare with an explicit confounder correction? Like if you just include PCs into your analysis?
Right, yeah, so I guess for some of these methods, we're kind of explicitly modeling for these different confounders that are present in the data set. And as we can see, the performance in those cases tends to not do as well. And also, as kind of mentioned in kind of like the introduction to this problem, a big issue arises when a lot of these confounders are hidden. And so you can't really explicitly model for them.
Well, but what if you just include like the principal components?
Oh, I see, sorry.
So you do some kind of naive or complicated hidden confounder method.
Right, yeah, so I guess I don't think we've tried that yet. So yeah, perhaps kind of incorporating those principal confounders might also lead to kind of a similar type of performance. But I guess for now, no method that we know of has been made to kind of demonstrate this. So we, for now, haven't benchmarked it with our method.
Thank you.
Thanks for the talk. I have a quick question on how do you select the quantiles? Like it seems for extreme imbalance data, you need to select a more stringent quantile, the other word. But is there a data-driven way that given the data set you have, automatically select a quantile?
Yeah, so the way we select quantiles is actually based on the data. What we do is we essentially permute the case control labels, where we're assuming that under permutation, we shouldn't see some sort of signal. And we're driving up the quantile until the kind of p-value that we're calculating based on the beta distribution that I described is below 0.5 or whatever metric that we're using. And so this does tend to lead to pretty stringent quantiles that are near the maximum function, but they tend to be at least slightly lower, which leads to an increase in power.
Okay, let's give Sam another round of applause. Okay, so next up we have Gong Li, who will be talking about pseudo-time analysis with single-cell sequencing and imaging data.
Hello, everyone. My name is Gang Li. I'm a postdoc at Bill Noble's lab here at UW. Today it's my great honor to present our recent work, pseudo-time analysis for time series single-cell sequencing and imaging data. As yesterday, we have seen many talks talking about cells are growing asynchronously and also cells differentiate at variable states. Here, I show you a very simple example for a mouse embryonic stem cell differentiation process, where cells differentiate to embryonic body cells and all the way to the neuroprogenitor cells. And imagine for a cell, maybe at day 7, you have growth and transit to embryonic body cell, but even another cell at the same time point may grow slightly faster than this cell, and another cell maybe grows slightly slower than this cell. When you combine all the cells at the same time point, look at their cell time, you might see a unimodal distribution. And when you have a time series single cell and single cell data set, you might have a multimodal pseudo-time distribution. And the goal of our project is to infer the pseudo-time for such kind of time series single cell data sets. And the microscopic image coming from the Bonora et al. Genome biology paper. So once you have a pseudo-time for a time series single cell data set, here is a gene by cell matrix. Each cell have a time label. You can use them to identify some time variant genes, the genes that are changing along the pseudo-time trajectory, and also you can order your cells based on the pseudo-times. We are not the first one to tackle this problem. People in the field first try the principal components. Usually you'll find your first principal components roughly correlated with the time label you have. Here in this pancreas cells, there are 8 donors. The age of the donor ranges from 1 to 54. You find the first component roughly correlate, but you also find a lot of the models are kind of overlap with each other. And more, there's another method called monocle. If you in first time, you will might heard of this before, but as this example, you will see one of the monocle component is correlated with time, but they put the age 21 on the rightmost, which puts basically the first component doesn't quite reflect the true time label we have of the single cells. So we found that for those unsupervised methods, their first component usually are driven by other global variability. The temporal variability might at the second or the later components might be different from the global variability. Based on that, recently there is a method called supertime, which adopt ordinary logistic regression, which explicitly use a time label for each cell to train a penalized ordinary logistic regression to select a subset of the genes and to infer the pseudotimes using the time labels. Here is a Caroline illustration where you have a simple score function. Here you have 5 time labels. Supertime will infer 4 threshold for this score function and classify the first group versus the rest, the first two groups versus the rest, so on and so forth. However, when they apply to the same data set, although the performance is much improved compared with PCA and monocle, we still see a lot of overlap in this pseudo-time model distribution. We motivated by this limited classification accuracy, and also the pseudo-time inferred by supertime is rank-based. You see the range of the pseudo-time on the x-axis is from -10 to 15. Doesn't quite reflect the true donor ages. So based on that, we propose a new framework called Skeptic Single Cell Pseudo-Time Classifier, and which basically adopt a multi-class support vector machine model to first infer the class conditional probabilities for each cell, and then infer pseudo-time by taking the conditional expectation. And compared with the previous model, we improve our predictive power using not just one score function for all different classes. We will adopt k different score functions. And also our Skeptic model would allow for nonlinearity when you consider, say, reader-based function, kernel function to generate nonlinear decision boundaries. The third difference is for the supertime is showing your training performance on the whole data set, where we propose to do nested cross-validation to provide a more robust and prevent the overfitting on the training data set. What we do is, like, for each data set, we for each fold of the data, for example, the yellow test fold, we will train a model on the purple set and predict the pseudo-time on the yellow set, and repeat this for the all the data set we have. And we first test our model and compare with SuperTime and mouse embryonic stem cell differentiation data. On the left is a confusion matrix for our skeptic model. On the right is a confusion matrix for SuperTime. A higher number on the diagonal indicate better performance of our classification model. And in this comparison, our model achieve a 94% accuracy compared with SuperTime slightly less than 90%. And the improvement is significant according to a Fisher exact test. And we also want to point out SuperTime does not have a train test split. It's simply the training performance. So despite the bias towards SuperTime, we still outperforming them. And you might be curious if our skeptic model are simple learning batch effects or the real temporal structure, because imagine a scenario for each time label, if you have a strong batch effect specific for that time label, you can still train skeptic achieve a perfect confusion matrix look like this. To confirm that, we perform a leave one timeout analysis. So every time we mask, for example, the T1 time and train a model on T2 and to T5 and predict all the cells from T1. So in a scenario, if the model are simply learning batch effects, we expect the model to predict uniform errors on the off diagonal. So in this case, since we mask the time for each time point, so the diagonal are all 0. And if the model are truly learning some temporal structure, we will expect a successful model to predict time points to be adjacent time point. So if we mask T1, we'll expect the model to predict most of the cells to be T2 instead of the rest of the later 3 time points. And when we do this leave one time point analysis, we found indeed our model are predicting the most of the cells close to the diagonal. And this is third time. And what is a little bit different is that we don't, for the day 11 cells, we don't predict too much on the day 21. This is because the middle 3 time points are embryonic body cells. So they looks, for the day 11, they looks more similar to day 7 instead of the day 21 neuroprogenitor cells. And on the right-hand side, the pseudo-time visualization instead of confusion matrix. After test out on this data set, we next benchmark our skeptic model on 6 public single-cell RNA sequencing data set. 5 of them evaluated in the original SuperTime paper, which they have shown to outperform PC and molecule. So we directly compare their performance on those 5 data sets. And on all the 6 data sets, our skeptic model achieved uniformly better performance in terms of the classification accuracy, and the improvement is also significant according to Wilcoxon rank test. After showing that the skeptic model can work well on single-cell RNA sequencing data, we next test can we generalize this model to single nucleus imaging data. Our collaborator Hunjin from Dr. Fuller's lab here at UW, they help us to collect the same 5 time point differentiation mouse embryonic stem cell differentiation data, but have the single nucleus image. So here I show you some random crops of the image from 5 time points. And specifically, instead of running on 2D image itself, we adopt image processing pipeline, which using the pre-trained residual net to embed all the 2D images to a 1024 dimensional representation. And after we have the 1D vector for each image, we run the skeptic model and compare with SuperTime. Again, our model can achieve improved classification accuracy, and the improvement is significant according to the Fisher exact test. And more importantly, we find the skeptic model can provide a more fine-grained label compared with the original time point label. For this, we compute the absolute correlation of the pseudotime for each cell with some morphologic features. Here, I show 2 of them, nuclear area and nuclear parameter. We find skeptic pseudotime have a higher absolute correlation. This actually are supported by the literature that during the mouse embryonic stem cell differentiation process, the embryonic stem cell will have large nucleus, and for MBC, they will have smaller nuclei. After showing the skeptic model can work well on the image data, we further test on the sex differences on both single cell attack and single cell RNA sequencing data set. So here I show you the male single cell attack sequencing results from skeptic model. So they don't have the 5 time point, only 4 for this one. But when we compare it with the female cell performance on the single cell attack, for all the cells of day 7, if we look at the attack pseudo time from skeptic model, we find the female cells have a heavier tail on the left-hand side. This actually is because during the embryonic stem cell differentiation, the female cells show some delayed differentiation pattern, which is well known in the literature. We did the same analysis for the single cell RNA sequencing data set by comparing all the RNA pseudo time for the day 7 cells. We observe a similar delay pattern in the RNA pseudo time. And last but not least, we have also tried to extend our framework to the single cell high-C data. For this purpose, we extract the contact decay profile for each single cell high-C data, and then run our skeptic model. We get a pretty decent accuracy. And here are the third time for the 5 time point. You see those models are not very overlapped with each other. Hopefully, I have convinced you our skeptic model can outperform the state-of-art method SuperTime in both time series single cell sequencing and imaging data. And we have shown that skeptic can work well for various types of the data set. And it can detect sex-specific differentiation from single cell attack and sequencing data. In our manuscript, we also show that we can identify some methylation delay using some co-acid data, but with time limit, I will skip this. And our code is available. The manuscript is also available on the bioarchive. With that, I would like to thank my advisor, Bill Noble, and Christine and Xinxian for providing us mouse embryonic samples, and Doc Ford's lab for generating the images for the analysis, and Jen Philip for the machine learning expertise, and all the Noble Lab members. And thank you for listening. I'm happy to take any questions.
Great questions for Gong?
Hi, thank you for the super cool method. I was wondering if you could provide any like intuition based on the formulations why your method performs better than SuperTime.
Yeah. Yeah, so there are 3 major motivations. The first one, the logistic regression is only 1 score function with 4 threshold. And for our multi-class SVM model, for each classification, you will have a score function. So that greatly improves the predictive power. And also, logistic regression model is a linear-based model. And with RBF kernel, we can have nonlinear decision boundary, which also outperform the traditional ordinary logistic regression. That's a major motivation.
Thank you.
Yeah, I was just wanted to ask, how would this method compare to like other pseudotime inference methods that try to map out like a continuous time label rather than like these discrete time labels that you use for your model? I was wondering if there's like an advantage over using septic over those kinds of methods.
So just want to clarify, our model have both like a discrete label and also the continuous label. For example, I have shown a plot like this. We have a continuous pseudo-time inference by conditional expectation. So we did not compare with other methods like PCA and Monaco because in the previous evaluation, SuperTime have been shown that outperform other like pseudo-time inference methods. So we directly compare with, focus our comparison with SuperTime in this manuscript.
Nice talk. So I was wondering how your method does if you have very few time points. So if you have, obviously you can't do anything if there's only 1.
Yeah.
But if you just have 2, then I guess life gets more difficult, and like then how the comparison to Monaco and that sort of approach.
Yeah, that's a great question. So for the time series data set, at least we request at least 3 time points. But when there are only 2 time points, you can still train a classification model. But I'm not sure. We haven't evaluated on such data set, but that's very interesting to look at them.
Feels like in that setting, there should be something that's like in between what you're doing and
Yeah.
what can look at the
Yeah.
trajectory. Thanks.
Thanks for the suggestion.
All right, let's give Gong another round of applause.
Kind of moderate the time and questions. Yeah, exactly. I think one person just figures it out and do it. Yeah. Yeah. Okay, so then share screen and then wait. Share screen. So you want to do the first presentation.
Understanding the fact
You sure it's fine to leave?
Yeah, yeah. On YouTube, is it fine now?
Okay.
Because here's presenter view. That's right, but what is on YouTube? Can someone check?
Yeah, check. This is what it looks like. Okay.
Oh.
Yeah, you can see.
Okay, so this all works. And then if I want to switch it, I just what do? I stop sharing and then share the next one.
Yeah.
Yes.
So it's like, so you tips you like, you're a hundred thing hybrid. Whether all these working my background is from actually So I said my background is kind of so I'm yeah, I guess that makes it more difficult. Yeah.
And so I wanted that was really basically there. Is that like each you like propagated signal out with like sites, but not around. And then like yeah, inside and out with the lab. And then just like base there. So you go to the lab again, you don't have lab yeah. Right now,
Yeah, I do
Is the microphone delivery my chat too?
Yeah.
Yeah, okay. Where's the yeah. Oh, yeah. Let me just close it. Where is Zachary Sims?
I think that's
Oh, him. Okay.
I'm not sure, but he'll come up.
Yeah. You're going to introduce the session, or
No,
you
I'm
want
just,
me to?
yeah, I'm just gonna set it up. and
Yeah.
do the talk.
Let me just, just to
Yeah.
gather attention. Everyone, welcome back. Okay, so in the next 1 hour session, we are going to have 3 amazing short talks. I need to see the title of the first one. Okay, are you ready, Sarah? Okay, not yet. Okay. Okay, thank you so much. Welcome back.
Some technical issues.
What's going on? It's because of the hybrid setting, I think. How was everyone? Good? Good? Raise your hand. Okay. Almost there. Okay, welcome back, everyone. So one of the three amazing talks in this session will be about a masked image modeling approach and presented by Zachary Sims from the Oregon Health and Science University.
Hi. So yeah, my name's Zach. I'm from OHSU. I'm a research engineer, and I'm here with my PI, Youngwon Chang, here to talk to you about our efforts in producing a machine learning method that can address the limitations and potentially democratize multiplex tissue imaging by learning to generate tissue stains in silico. So to set up the problem, multiplex tissue imaging is this emerging family of technologies that is really powerful because it allows us to do this single cell analysis within the spatial resolution of tissue. And so the technology that I'm going to be talking about today is cyclic immunofluorescence. And so this was a multiplex tissue imaging technology first proposed at Harvard Medical School. And so it involves this iterative process of 4-channel imaging along with the iterative cycle of 4-channel imaging with and then followed by bleaching to remove the signal, and then repetitively doing this with different markers each time. And so you can over successive rounds build up this panel of dozens of different markers that measure protein expression within a single sample. And so in the bottom left here, I show an example panel. So we call the set of markers that we're imaging here a panel. And so this is an example panel done on a colorectal cancer sample here. And so this is a 25 marker panel. And so you can see here that we can do it in eight rounds. And so in every round, one of the channels is reserved for DAPI, which is a nuclear marker. And so we need to have this marker in every round so that after we've done our imaging rounds, we can align and register the different rounds so that we can do our single cell analysis. But then the other 3 channels are allowed for different markers of protein expression here. And so up here, again, is an example of an image of a 10-round cycle of SciSif with the 3 markers in green, white, and red, and then DAPI in blue. And so the most common analysis that you do here after you've done the imaging is to segment each cell and then use the cell boundary for each individual cell to measure the average pixel value for every marker channel in your image. And that gives you a mean intensity value for every marker for every cell. And so you can gate that value, and that will give you a classification of a positive or negative cell for that marker. And that's how we can do cell phenotyping within a spatial resolution. And so this is a really powerful approach to investigating the tumor microenvironment and has a lot of potential applications for improving our ability to perform diagnosis in a pathology setting. But unfortunately, there are limitations that we have to address. So within SciSif, the main limitations here is time. So one of the main limitations is time. So the number of rounds that you can do is limited because one round takes up to 1 day to complete. And so additionally, as you start doing more and more rounds, the number of staining rounds that can be conducted is limited by tissue quality, which degrades in later rounds. So you can see up here, for example, in later rounds, you start to get tissue loss, and that means that certain cells are literally just not going to show up in these rounds. And so markers that were stained within this round, you essentially have a data missing this problem where you're not going to get markers for that cell, or those markers for that cell, yeah. And so then in commercial multiplex imaging systems that are more commonly used, the number of channel is limited because you can only do single round staining. And so you can only do about 6 or 7 markers in that setting. And so if you wanted to have a higher plex system, like Rareside Orion can do 17 or 18 markers per round, but those require special machinery, and that is cost prohibitive. So in order to proliferate this technology, we want to be able to overcome these barriers. And so we look to a software solution to potentially address these issues. So to formulate this machine learning problem, we essentially propose this two-step problem. So the first is, can we take our full panel of markers and identify a representative subset of markers that retains the full information of the full panel? And so we call this step the panel reduction step. And then so once we have a candidate reduced panel, then we want to be able to learn a way to impute or reconstruct the full panel and the markers that were withheld, essentially. So if we can do this, then we can have three immediate use cases. So the first is we can address the data missingness problem of where there's tissue loss in later rounds. We can impute those stains and address the data missingness issue. And then the second use case can be the potential to democratize higher plex systems by taking lower plex systems and synthetically expanding them to higher plex systems by conducting the staining in silico. And then thirdly, we can also help to better optimize and design better panels by, if we can identify easier to predict markers, then we can leave those markers out of the actual staining and imaging, and we can replace them with more difficult experimental markers that can potentially identify more rare cell types. So this is an emerging problem that has a few different solutions that have been proposed. So our lab was the first to publish a method for panel reduction and marker imputation done by a former grad student, Luke Turns. And so this method is a two-step process. So first we have to identify an optimal panel, optimal reduced panel, by finding the subset of markers whose mean intensity are maximally correlated to the remaining withheld markers. The reduced set of markers is then fed into a multi-encoder VAE as a multi-channel single cell image, and the full cell image, and the reduced set of markers is then used to reconstruct the missing channels that correspond to the withheld markers. And the next method was proposed by a group at Stanford that is a three-step process. First, a concrete autoencoder is trained to learn a representative subset of 7 markers. Then a multi-scale representation of each cell for each marker is fed into a ResNet to learn morphological features for each cell. Then those features are fed into a regression model to learn the mean intensity value of each withheld marker for each cell. The third method in this family of methods is from Carnegie Mellon, and they propose an iterative approach to learning a reduced panel by training a UNet to reconstruct image patches where a single marker is added to the reduced panel at each iteration, starting with one input marker and continuing until the target reduced panel size is reached. So the most recent method is from a group mostly from Harvard that uses a UNet to reconstruct patches of a single marker using 6 input markers. So the first two methods that I mentioned are most comparable to our approach, and this Luke Turns implementation will serve as our baseline for my method that I'm discussing here. And so they're most common to us because they use single cell images as input, whereas the second two methods use an image patch containing multiple cells as input. So all of these methods except the Sun et al. Method suffer from the same limitation. And that is that the reduced panel is selected prior to and independent of marker imputation. So that results in a potentially asymmetric pair of problems where the identified reduced panel is not guaranteed to be the optimal panel for marker imputation. So, and additionally, all 4 methods suffer from the limitation that each trained model is only compatible with one reduced panel. So that limits out-of-the-box use cases. So the framework that we implement our panel reduction marker imputation process through is through using a clever implementation of the masked autoencoder. So we've talked a lot about over the past couple of days about masked language models, and so the masked autoencoder is this extension of masked token prediction to images, where the 2 key differences here is that we use an encoder-decoder framework where the encoder only sees the visible image patches, and the decoder then will take the latent representations of those visible patches along with the mask tokens, and then use to regenerate the missing patches. And then it's trained with mean square error on the pixel values. And so if you have a standard RGB image, then you're going to be masking along the height width axis so each token in your input is going to be like, for example, a 16 by 16 pixel by 3 color channel image. And so that's how you represent a sequence within this context. And so cleverly, what we can do here is simply mask along the channel axis for our multi-dimensional images. So what we feed in as input are these single cell images cropped to a 32 by 32 pixel region, and then they have the 25 channels for each marker stain. And so conveniently, 25 can be the 25 markers can be reformatted into this 5 by 5 grid, where essentially each patch now is a single image channel, and we can just mask channels the same way that we were masking patches in the original implementation. And so the original authors show, as opposed to BERT, that a high image masking ratio is effective here. And so they do about an 80% masking ratio, and that allows us to compress the information of this image, the semantic information of this image, by 80%, which is effectively what we want to do here, is compress the information of our multi-channel image into a subset of markers. So to bring it to my implementation, how we use this. So first, our data set consists of a single breast cancer tissue microarray, which was collected under the auspice of the Human Tumor Atlas Network. And it contains 88 cores of 6 different breast cancer subtypes. And so we segment this TMA, and we result in a data set of over 700,000 cells. And so we crop to the 32 by 32 pixel ROI and zero out the background. And then we randomly mask 50% of the input channels during training and train the model to reconstruct the masked channels using mean square error as the loss function. Because we randomly mask channels during training, our model has generally learned to impute any marker from any other marker in our training set. So additionally, the transformer architecture allows us to utilize pre-trained weights on variable input sizes. So even though our model was trained to predict 13 markers from a panel of 12 randomly chosen markers, we find that during inference, we can change the number of input markers, and the better prediction performance will result accordingly. So as we increase the amount of input markers, we'll get a better prediction for our remaining withheld markers. And so we seek to leverage this by using our train marker to identify optimal reduced panels. And so this has the benefit of reducing the computational complexity by only having to train one model, and also has the benefit of unifying the panel reduction and marker imputation steps into one model. So how do we leverage this? So it's still intractable to evaluate every subset of markers for every panel size. That would result in over 30 million different combinations to experiment with. So instead, we propose a greedy selection process of finding the next best marker to add to the panel. And so to conduct the panel selection process, we can progressively increase the number of unmasked channels input to the trained model, starting with the best 1 marker panel and ending with the best n-1 marker panel. At each step, we find that the next best channel to be unmasked by choosing the channel that results in the best reconstruction of the masked channels, which we evaluate based off the Spearman correlation between the real and predicted mean intensity values. So this can be interpreted as choosing the most informative remaining marker. This process only requires us to evaluate 300 different candidate reduced panels for our data set of 25 markers. And so this example on the bottom left here is showing a toy data set of 4 markers. And so we first start by fixing the 1 marker panel to be DAPI, because we always need DAPI to conduct the later downstream analyses. And so we then iterate through the remaining 3 markers to find the best 2 marker panel and select the next best marker. And then we include that into our reduced panel size, and then iterate through the remaining 2 markers to find the best 3 marker panel. And so, yes, that clarifies. And so this approach results in a significant performance increase over our lab's initial approach, as well as streamlines the process. Here we compare between the MEVAE model and the MAE model on the same breast cancer TMA data set. And we evaluate the Spearman correlation between predicted mean intensity values for the withheld markers using 6 different reduced panel sizes. The yellow dashed line represents the performance of the MAE model on the panel size of 3, 6, 9, 12, 15, and 18. And the yellow violin plots here represents the performance of MAE using the same panels. And then the green violin plot here represents the performance using the reduced panel selected via iterative selection that I just demonstrated. So the other advantage of our MAE approach is the ability to evaluate any panel size with the same model. Since a new model must be trained for each panel, in our previous approach, analysis was only done on a fixed number of panel sizes. So I have, for example, here the optimal panel identified for 3 markers in our last implementation, and then 6 markers here. And so the top table shows the reduced panel selected via correlation. You can see that the 6 marker panel consists of removing ECAD and adding Cyclin, D1, Vim, and ER. The bottom table then shows our panels selected via iterative selection. And not only is it easier for us to evaluate more reduced panels, you can see that each reduced panel is a direct subset of the next panel, which greatly increases the interpretability of our panel selection procedure, which I will detail in the next slides here. So we can get a very interesting look at what's happening as we add markers to our reduced panel here as an example. So we first start by fixing DAPI as the first channel chosen on the right. The heat map indicates the Spearman correlation of each predicted mean intensity value for the marker, and the chart on the left shows the mean value of each column on the right. Using just DAPI as the reduced panel results in a poor reconstruction of the remaining 25 markers. We then iterate through the remaining 24 markers to find the best marker that can reconstruct the remaining 23 markers, and we find that EGFR is best. On the left, you can see that the prediction of the withheld markers has significantly increased, and on the right, the change in color of the pixels in each row demonstrates the improvement in the reconstruction for that marker when EGFR is added to the reduced panel. And then in the next step, this is where it gets interesting, is that we iterate through the remaining 23 markers and choose CK19. And then we can see that on the right, the reconstruction of pan-CK has significantly increased with the inclusion of CK19 into the reduced panel, which makes sense since pan-CK and CK19 are highly co-expressed. So when we do this for every panel from 1 marker all the way up to n-1, then we get this nice little pyramid that with this red vertical line, we can slide it left or right based off our own personal threshold as to how acceptable of an error we would like to tolerate in choosing our reduced panel. And we personally find that a 9 marker panel works well and results in a 0.87 mean Spearman correlation on the 16 withheld markers. And so this scatter plot is showing the real versus predicted mean intensity values for each marker, where each point on the scatter plot represents a cell here. And that's also shown here for 3, 6, 9, 12, 15 marker panel sizes. And as we increase the panel size, the prediction of the intensity values increases greatly. And so we also show this works on a colorectal cancer data set, and we're still working on investigating multiple tissue types and working to integrate multiple panels from different tissue types, as well as panels that have different markers on them. So that's our future work. And so I would like to thank my lab members at the Quantitative Bioimaging Lab at OHSU and our collaborators at Harvard Medical School, along with our grants funded by the NCI, the CUNY Foundation, and here's a link to the preprint with a link to the GitHub and model weights. And I'm now open for questions. Thank you.
Have you tested how important spatial information is?
So that is something that, no, we don't really incorporate at all, since we crop out the cells and then they're just input as a single cell. So we haven't necessarily looked at the spatial implications of this.
And have you looked at samples in different tissues?
So like I mentioned, we have, so in our previous work, we did the breast cancer TMA, and that served as our benchmark for this analysis. And we also have demonstrated it works on colorectal cancer, and that's sort of our future direction, is maximizing its performance and generalizability within colorectal cancer.
It just seems like something that might be very cell type specific or like organ specific.
Tissue, both because tissue types widely vary, right? But then also the marker panels that are designed for the tissues are often very different. So you don't have a lot of ability to have the same markers in different tissue types.
Hi, very nice talk. I was thinking about the use case of this. Are you thinking about having reducing the size of the marker panels for future tests, or because if things are missing in like real world scenario, it wouldn't be missing at random, right? You would have missingness that's not necessarily random. So how does it help to mask things at random from these panels?
Well, so the random masking procedure is useful because we then are basically ensembling a bunch of different random 12 marker panels to predict the remaining 13. So that allows us to kind of do this one-step training process, and that's how we can leverage it downstream to then do experimental different input panels. And then for the cell loss, I mean, I would assume tissue loss does kind of happen at random. I'm not sure if it's necessary. Maybe there is some sort of reason behind it, but sometimes you do just randomly lose cells, right? So that does solve that use case.
Thank you.
Just one more question.
Great talk. I was curious. So let's say that you have protein A that is very correlated with the localization of protein B under wild type conditions, but upon drug treatment, you completely disrupt the colocalization of protein A and protein B. If you've learned your colocalization model leveraging correlation from untreated cells, and then you want to see the effect of this on actual drug treatment, how do you feel confident that basically the correlations that you're seeing are going to hold upon different biological conditions?
Right. Yeah. So that's a great question. We don't consider perturbations or anything like that in our data sets. However, we are now working on including multiple samples into our training set, which has a wide variety of heterogeneity between samples. So, I mean, it's a hard question. I mean, our current approach is just ultimately to create a better training set that can hopefully represent the distribution of potential marker expressions. But you're right. I mean, if the relationships do fundamentally change, then our model would probably not work in that case.
Thank you so much.
Thank you.
Okay. So the next speaker is Yutong Wang from Berkeley. He's going to talk about the decoding cell-cell communications through deep learning. Yeah, please.
Hello everyone, I'm Yutong from the Song Lab at UC Berkeley. So the title of my talk is Decoding Cell-Cell Communication Through Deep Learning Reviews Novel Ligand Receptor Pairs from Transcriptomics Data. So I'll start with the motivation. So in order for the multicellular organisms to function well, then the cells, they definitely are not isolated. They have to be constantly talking with each other. So then the ligand receptor interaction is indeed central to the cell-cell communication. So what do I mean by ligand receptor interaction? So as we can see here in the figure, essentially the sending cell, they will be able to secrete the small molecules in green here. They're the ligand, and they will bind with the receptors on the surface of the receiver cells. Yeah, so this is how cells would communicate through the ligand receptor. However, it still remains to be a computational challenge to identify or predict the ligand receptor interactions that actually underlie the cell-cell communication mechanism. And this is especially important for G protein coupled receptor, GPCR. It's basically essentially just as the figure show here, it's a 7 transmembrane domain receptor, and it's actually also the largest family of the cell surface hormone receptors. This is a very important family of receptors because GPCR, they're actually targeted by 35% of the FDA approved drugs. However, actually, we have not fully explored the potential of GPCR yet. The reason is that approximately 20% of the human GPCRs still remain orphan. By orphan, I mean we do not know the endogenous ligands that will bind to these GPCR. So it's actually very important to be able to de-orphanize for these GPCR in order to better understand the signaling pathway and also potentially to identify the novel drugs. So yeah, that's here the biological question we want to answer, how to identify the unknown ligands for the receptors. Okay, so in order to answer this question, here we turn to the principle of spatial proximity. So the cell-cell communication is the signaling mechanisms of cell-cell communication can be classified based on the distance. So here on the top, we can see that's autocrine action. Essentially, the small molecules secreted by one cell will actually act onto the same cell itself. And then the second type is called paracrine action, where the ligands will act onto the nearby cells. Last but not least, there's also the endocrine action. Essentially, the ligands will act on the distant cell, and normally the circulation is through like bloodstream or something like that. So due to the localized autocrine and paracrine effects, so normally, or most of the time right now, inference cellular communication often relies on leveraging the spatial proximity from spatial transatomics dataset. So thanks to the very nice illustration from the two keynote speakers about spatial transatomics, I think, hopefully we all have a very good understanding about spatial transatomics right now. And here, just to briefly recap. So here I'm showing the data of spatial transatomics data of 4 different genes. Essentially, for each gene, the X and Y axis that correspond to the spatial coordinate in space. And for each pixel here, for each dot that correspond to 1 pixel, where each pixel contains like normally like 1 to 10 cells, multiple cells, and the color for each pixel that correspond to the spatial gene expression for this specific gene in that specific spot or pixel. So this is spatial transatomics. And then, so essentially, why can we leverage on the principle of spatial proximity? This is because, as what we're talking about, because of the autocrine and paracrine effect, so the cells that are close to each other are more likely to interact. So this implies that if 2 genes, for instance, like the ligand receptor pairs, if they are indeed interacting, then it's more likely that they will share some very similar spatial gene expression pattern. So for instance, as we can see on the top here, for these 2 genes, their spatial gene expression pattern or the spatial distribution is very similar. And indeed, in the actual biology, they are indeed interacting. Notice that here, the interaction, that's not inferred based on the spatial transatomics. That's the underlying ground truth. And so this is not like the bottom pair, where for this pair of ligand receptor, they're actually indeed not interacting and they do not share a similar spatial distribution. Okay, so what are some of the existing methods that are trying to solve the problem? So the first big category include, for instance, cell phone DB or cell chat. Essentially, they're trying to curate the known ligand receptor interactions from the literature. However, the problem with that is that they leverage on the prior knowledge and they're not suitable for predicting the novel interaction. Actually, I'll also show some of the results if we try to predict the novel interaction using them. The other is spatial DM that was published earlier this year. Essentially, they're actually having similar methods like this. Essentially, what they're trying to do is they're trying to leverage the spatial statistics, for instance, such as spatial cross-correlation like Moran's eye, to try to identify the co-expressed ligand receptor pairs. However, the problem with the spatial statistics is that they lacking capture like the complex and also nonlinear spatial patterns. Last but not least, there's also GCNG, which is a kind of graph neural network that's trying to solve the problem. However, the problem with that is that, so even though there are like 20,000 protein coding genes, but there are only like hundreds of ligand and receptor genes like encoding the ligand receptor. And this only includes about 1,000 to 2,000 true interactions. So for this kind of graph neural network, because they rely on supervised learning, but they also have very limited label data. So this is actually very hard for them to truly make use of the spatial transatomics data set from like 20,000 genes. So, okay, now we have our question. How could we try to come up with a method that can predict the novel interaction, and also it can capture the nonlinear spatial relationship. And most importantly, how could we learn the useful representation from the massive unlabeled spatial transitomics data? Okay, yeah, so here, what we're proposing is basically a two-step method, where the first step is that we want to treat the gene spatial expression as an image for unsupervised feature extraction. What I mean here is basically for the spatial gene expression of each gene, we take that as one, we consider that as one image, and then we just pass the 20,000 protein coding gene images through this convolutional autoencoder. And then the task here is just trying to reconstruct the images. This will help us to learn the latent embedding space for each gene. So, okay, after we get the pre-trained latent embedding for each gene, then we can move on to the second step. That's for supervised classification. So essentially, okay, as we can see from the left, so we can feed the model with a pair of the pre-trained latent representations of one ligand and one receptor. So we have each of them to go through one subnetwork of this Siamese neural network. Essentially, what Siamese neural network is trying to do is that, so basically we have this twin, a pair of twin subnetwork with shared model architecture and model weights. This is trying to encourage our model to be able to do similarity modeling. Okay, so after we have both the ligand and receptor go through this subnetwork, then we aggregate the output, and then we just run that through some simple binary classifier in terms of whether for this specific pair of ligand receptor, whether they interact or not. Okay, yeah, so that is our two-step approach where we can make use of all the 20,000 protein coding genes, not only including the ligand receptor corresponding genes. So our method is called GG pair, and indeed it's performing much better compared to other state-of-the-art. So what are we trying to do here, or what is the experiment? So the data that we're using here is a mouse brain spatial transitomics data. And so essentially we're performing a 5-fold cross-validation. So for all the receptor genes, we split that into 5 different folds. And then we take, each time we take the 20% of the receptor genes as testing, and then the rest of the receptors as training. And then for the ligands, we can just, this is because, so here we're splitting the receptors for training testing splitting because we want to prevent data leakage. And then for the ligand, we can just randomly match with the receptors. And we repeat this 5-fold cross-validation using 20 random seeds to ensure robustness. And so as we can see here, so basically the metric that we're using here is the area under the precision recall curve. And on the x-axis, the leftmost correspond to all the receptor genes. And then we stratify based on different receptor families, including this GPCR family that we mentioned at the very beginning. So our method is called GG pair. It's to the very left. You can see that our model is performing much better compared to other state-of-the-art methods. Okay, and so focusing on GPCR, then we can do the next experiment as in, leave one out validation. So essentially what we're doing here is in each of the leave one out iteration, we extract one single GPCR receptor out as testing. And then we just pair this single receptor gene with all the possible ligands. So this will be our testing set. And then essentially, so then we'll be able to get the predicted probability for each of those pairs. And then we can rank all the ligands based on the predicted probability. So which is the x-axis that you can see here. So in expectation, we hope that the ground truth true interacting ligands will always be in the top ranked genes based on the predicted probability. And indeed, that's the case for our method GG pair. Most of the true interacting ligands are in the top 30 genes, but this is not true for other state-of-the-art. Okay, so in summary, we introduced this new method called G-gene pair, which is a novel framework for ligand receptor interaction prediction using spatial transatomics. And so essentially what we're doing here is like a dual deep learning approach with unsupervised convolutional autoencoder for spatial feature extraction, and then followed by a supervised Siamese neural network for interaction classification. So our method outperforms the existing methods, enhancing the understanding, especially for GPCR signaling, which is a very critical venue for the drug discovery. And I also want to acknowledge my colleagues, Fending and Antoine, on this project, and also my advisory. Okay, yeah, with that, I'll be happy to take any questions.
Thank you. Do you have any rationale of why the ligand receptor interaction would be non-linear, and maybe one or few example that like the simple linear geostatistic methods fail to discover those pairs, but like it could be several, you know, your deep learning model will be able to predict well?
Yeah, well, okay, essentially in terms of the linearity, that's just based on the Euclidean distance, right? But I mean, in terms of the actual tissue, in terms of the communication, or in terms of how the different cell states within the tissue, they might be spatially distributed, that might not be linear, which is why it's better to consider that as like a complex image.
Ligand for predicting cell ligand receptor interaction, right? So
Yes.
there is something other than co-expression. Then I was wondering what would be that thing? Like what's the biology of those like non-linear, like not co-expressed ligand receptor interaction?
Yeah, okay, so I guess, yeah, your question is in terms of this part. So so first of all, like in terms of the actual ligand receptor interaction, I think there could be like actually multiple different steps in terms of the whole signaling pathway that might be very complicated. And also in terms of here, I think we're not saying that spatial co-expression is not helpful at all. We're just saying that in terms of the co-expression, it's not sufficient to only consider the linear co-expression. As in, if we only use the spatial statistics, like more like you can consider that as like Pearson correlation in the spatial sense, then that's not sufficient. That's what I mean. Okay. Yeah.
Okay. Oh.
Thanks. Nice talk. I wanted to understand the autoencoder setup a little bit more. So is it you have multiple images, or each gene effectively counts as like one sample, and then it's one network for every, like, shared across all genes?
I see. Yeah, it's like one network shared across all the genes.
Okay, so you just need one image, because then the genes are kind of like your samples in how we would normally think about training.
Are you saying that each gene can be a channel? Is that what you're saying? Or
No, well, it could be, but that's not what you're doing, right? Because
Yes.
this is just one channel. And so
Yes.
your sample size for training this is like 20,000 or whatever the number of genes is.
Right.
Okay.
Yes. Mhm.
Okay, thank you very much. So the next speaker is Mu Qiao from LinkedIn. He's going to talk about deciphering the genetic code of neuronal type connectivity.
So yeah, first, thanks to the organizers for this wonderful conference. And it's exciting to learn all this excellent research. So my name is Mu Qiao, and I was trained as a computational neuroscientist, but now I'm a machine learning practitioner at LinkedIn. So today I want to share the research about applying algorithms stemmed from recommendation systems to this problem of neuronal type connectivity. So I want to start by asking the question, why is neuroscience research challenging? Well, there are many reasons. A clear observation is that there are many different types of neurons, and there are complex synaptic connections between them. The complexity is evident even in this small piece of tissue within the central nervous system called retina, which is at the back of our eyes. The retina is an excellent model for neuroscience because of this regular arrangement of retinal neurons and their synapses. There are five classes of neurons in the retina: photoreceptors that detect light, and interneurons, including horizontal cells, bipolar cells, and amacrine cells that relay and transform the signals from photoreceptors. And finally, we have retinal ganglion cells that send the axons to the retinal recipient areas. There are over 70 different types of interneurons and over 40 different types of retinal ganglion cells. And each specific type of retinal ganglion cells form specific connections with certain types of interneurons in this synaptic layer called the inner platform layer. And the consequence of this connectivity is that each type of retinal ganglion cell is tuned to a specific feature, such as local motion, approaching motion, lateral motion, or color contrast. This knowledge is abstracted in this wiring diagram of what we call neural circuits, which we use to model and explain specialized encoding of, for example, visual features. However, despite this wiring diagram, the genetic underlying of the specific connectivity, especially that in many other parts of the brain, is still largely unknown. Part of the challenge here is the limitation in traditional approaches, which involve genetically manipulating the expression of candidate molecules in specific cell types, and then evaluating the effects on their connectivity. They are inefficient and inadequate for the following two reasons. First, neurons often have complicated connections, with one neuron potentially linking to different types of other neurons. And the second, the genetics behind these connections can be intricate. Instead of having one single molecule mediating the connections between two neurons, it's possible that gradients of molecules or a combination of different molecules that mediate specific neural links. These challenges ask for a systematic approach to decode neuronal type connectivity. Fortunately, we now have emerging data from single-cell transcriptomics and connectomics that pave the way for this decoding effort. The single-cell transcriptomics provides us detailed gene expression profiles of individual neurons, which are then clustered and classified into neuronal types. On the other hand, we have connectome, or anatomic reconstruction with electron microscopy. From this anatomical reconstruction, we not only can reconstruct the morphology of individual cells, but can also visualize individual synapses between neurons. So this provides us detailed connectivity matrix between neuronal types, such as the one between presynaptic bipolar cells and the postsynaptic retinal ganglion cells in the retina. With this data, an intriguing question arises. Can we predict neuronal connectivity based on gene expression? If so, then we can look into the gene expression data and identify which genes are critical contributors to this prediction. So this question parallels one addressed by the recommendation systems in digital platform, particularly in this product, people you may know, which you probably have encountered on LinkedIn or Facebook, where connections between users are predicted based on their profiles. So let's see how this is solved. In general, a recommendation system suggests an item, such as a movie, characterized by a feature vector y, to a user whose feature vector is x. It does so by projecting the user and item features into a shared latent space, where similarity in the space predicts user-item interactions. A common algorithm for this is called collaborative filtering. Let's illustrate this with this simplified example. Suppose we assign to each movie a scalar from -1 to +1 that describes whether the movie is for children or adults. And we also assign a scalar to each user from -1 to +1, reflecting the user's interest in children's movie or adult movie. Then the dot product of the movie's embedding and the user's embedding should yield higher value for movies that align with the user's preferences. On the right side, a checkmark identifies a movie that a particular user watched. You can see that the third and fourth users have preferences that are well predicted by this model. However, using only one dimension embedding, we cannot predict the user-item interaction of the first two users. However, when we add the second latent feature, the degree to which the movie is a blockbuster or an art house movie. We can now represent each movie with the following two-dimensional embedding. Now the user-movie interactions are well explained by the similarity of the latent features, and we can even predict that the last user is unlikely to watch the movie Shrek. Although here these embeddings are hand-engineered for illustration purposes, in a real-world application, these embeddings can be learned automatically, which is the power of collaborative filtering. So formally, assuming that we have access to the ground truth interaction Vij between a user I and item J, then the learning task is to minimize discrepancy of the predicted interaction and the ground truth. Expressed in the matrix form, we are minimizing the Frobenius norm of the residual matrix. Now the question is how to adapt the bilinear model to neuronal type connectivity. By considering presynaptic neurons as users and postsynaptic neurons as items, we can now predict synaptic connections similar to predicting user-item interactions. In an ideal scenario, if we know gene expression and the connectivity of each cell simultaneously, this translation becomes straightforward. The only difference here is the inclusion of this weight matrix adjusted based on the cell counts in each presynaptic and the postsynaptic neuronal type. This is essential, particularly in unbalanced datasets, where a few cell types could dominate the entire cell population. The weight matrix helps avoid model bias towards overrepresented cell types. In practice, the gene expression and the connectivity data are often from different sources. In this case, while we can align cell types based on established biological knowledge, associating individual cells is not feasible. To address this, we make a simplifying assumption, which is the connectivity and latent gene expressions of individual cells are represented by the average of their respective cell types. We then focus on minimizing the difference between the average connectivity and the gene expression of corresponding cell types, while imposing this constraint to minimize the variance of latent gene expression within each neuronal type. Essentially, the goal here is to minimize the discrepancy between these two sources of information, while maintaining consistency of the gene expression within neuronal types. To find the optimal transformation, we constructed the Lagrangian and used the alternating gradient descent method. Here is how it works. In each iteration, we keep matrix B fixed and adjust matrix A by moving in the direction of its gradient. Then we hold A steady and update B according to its gradient. This process repeats until they both stabilize at the solution. For this optimization, we select hyperparameters like the lambda and the dimensions of the latent space through a process of cross-validation. We tested our bilinear model with a retinal dataset of bipolar cells and retinal ganglion cells. Using five-fold cross-validation, we found that the best dimensionality of the latent space is 2. Then with these selected hyperparameters, the model successfully constructed the connectivity matrix between bipolar cell types and retinal ganglion cell types from the transcriptomic data. As I mentioned, the bilinear model of collaborative filtering is powerful because it can automatically decipher the latent embeddings. Therefore, we deep dive into each of the embedding dimension and see which attributes they encode. Interestingly, we find that each of the latent dimension contributes to a unique connectivity pattern or motif. To illustrate, when we group the bipolar cells or retinal ganglion cells based on whether they fall onto the positive half or negative half along each latent dimension, and then plot their stratifications using the red color or blue color, we found a clear distinction. For example, the first latent dimension separates bipolar cells and the retinal ganglion cells that synapse in the central versus the marginal region of the synaptic layer. In contrast, the second latent dimension focuses on the connections over the outer versus inner parts of the synaptic layer for both bipolar cells and retinal ganglion cells. Remarkably, these motifs align well with recognized properties of retinal neurons. Kinetics means whether the neuron have transient versus sustained responses, or polarity, meaning whether the neuron responds to light onside or light outside. Significantly, these motifs are not predefined or explicitly encoded into the model. Instead, they emerge naturally from the model, attesting the model's power to capture key aspects of retinal circuits. The inherent linearity of the bilinear model allows us to directly quantify the impact of each gene on connectivity through their respective weights. We can then identify critical genes based on the weight magnitude. The gene ontology analysis of these critical genes showed significant terms related to neurodevelopment and synaptic organization. But here I want to emphasize two sets of molecules. First, plexins A4 and A2. So these are receptors for semaphorin signaling pathways, and they are predicted to be important for synapses in the off layer. And indeed, knockout of the genes shows a disruption of the off laminar and its misplacement within the on laminar. And this happens for both plexin A4 and plexin A2. Another set of genes, contactin 5 or cadherin 8, known as cell-cell adhesion molecules, are predicted to be important for bipolar cells and retinal ganglion cells, synapsing in the on and off layers respectively. Indeed, previous studies provide evidence for their crucial involvement in shaping the on and off pathways. For instance, contactin 5 was shown to be important for the formation of the on pathway of this direction selective circuit. And the cadherin 8 expressed in this retinal ganglion cell type called GRG6 is important for its dendritic lamination in the off lamina. What's more important, it makes prediction on promising candidate genes that remain to be tested. Here, I want to pinpoint a set of genes called protocadherin. Protocadherin 7, 9, and 11X. They belong to the delta protocadherin family. Although their roles in the retina are not thoroughly explored, they show adherence in a combinatory manner and are important for neural circuits assembly in other parts of the brain. Therefore, they are promising candidates to be tested in the retina. So here, I want to give a brief summary of this talk. So we show that a bilinear model stemmed from recommendation systems was able to integrate transcriptomic and connectomic data to elucidate genetic underlying behind neuronal types. And we also show that the model's capability to capture recognized connectivity motifs of the retinal circuits and provide interpretable insights into gene signatures associated with these motifs. This makes this model a valuable tool for investigating gene regulatory mechanisms involved in neural circuits wiring. Looking forward, I'm excited about the future possibilities of developing a recommendation system AI for neuronal connectivity. Contemporary recommendation systems have the scalability potential to model the neuronal connectivity of the entire mouse neuron. As a comparison, there are about 2 billion daily active users on Facebook, while the total number of neurons in the mouse brain is about 70 million. What's more, the recommendation systems have advanced from linear models to deep models, such as the two-tower models that project the input features into the latent embedding through a deep neural network that can capture intricate nonlinear interactions of the input features, and to this deep and wide architecture that is a hybrid of the deep model and the linear model. And the purpose here is to balance the prediction accuracy, interpretability, and inference latency, and to recent practice of augmentation with large language models. I believe following a similar path, we will achieve a system that helps us understand the mechanisms behind the neuronal type synaptic specificity. Yeah, thank you very much. I'm here to take any questions.
Very interesting stuff. So the protocadherins have really interesting like alternative splicing and also alternative promoter choice that is known to control some amount of neuronal connectivity. So do you have any data where you can look at those aspects? It's obviously tough with 10x data. It's really hard to look at, but I don't know if there's other data sources you could try and look at.
Yeah, that's a very good question. Yes, so basically in this research, we were using available data, which is from 10x. But yes, I think also, yeah, someone also mentioned this, that this alternative splicing of protocadherins is important for this. synaptic connections. So yeah, I'm looking forward to integrate that data to explore this.
Okay, if you have a 20-minute break until 2, and then we'll have a panel here. So we'll set up some chairs for the live panel members will be here, and there'll be some online, and we'll do the panel.
My focus, wait, a little bit, so I I'm so far away. Oh. It would be cool. I think it would be exciting. You could imagine that this could be done better. Things what we can do with I think the idea of that position facial Yeah. Well, I guess the way they'd be like, so maybe you know, a combination of groups of people who would want everybody has one individual, but any combination of those groups of people is more or less likely to and then yeah, I do. I feel like sort of. I feel like I don't know. It's probably just feel like it feels like it's all right. But it seems like a thing that could be interesting to think about what you can do.
Yeah, you could imagine that the only then there's all this stuff like about the and then some of the ligands are dispersed over a very long distance. So like some of it is process and immediate, but some of it is like are access to like a status of one that yeah. Yeah, and then we have this page. I think that's more problem now. I think there's an argument there. Then we can use them to don't know all that. At least I heard it's not like I read about it. But I think the thing, I guess it's not that you can come up with like an arbitrary. Yeah, well, one thing we have to like, yeah, I guess I agree with you. I feel like there's two different assumptions. Like, get Also, that's part of my talk They've been doing like that here. Yeah. There's yeah, that's it. You have What's right?
Yes,
Table, yeah, right? James, can you hear us? Can you hear us? Oh, but that's one before.
Yeah, that's now.
Can you hear us, James?
I can hear you on Zoom.
Somebody said something?
I think.
Oh, Lina?
That's that. She can hear us.
Okay.
I think James might have to turn down his volume.
Yeah, I think James is muted, but I can hear you.
I'll just like briefly say we have this panel and then I'll hand it to you and then you and Elena can introduce yourself and then say something about the film. Is that right?
I don't know if James want to say something, but I think that sounds like
Oh, right. Okay.
Also, I can't currently turn my video on, so we should check if the panelists can turn theirs on.
Cannot. It doesn't allow me. It says the host has stopped it.
That's what it said before. Mine just changed, so now I can. I might change for you in a second.
Makes sense to turn on the closed caption just in case. Like,
No, not yet.
yes, because it's a little bit like muffled for me. A little bit larger by any chance.
Is there a full screen?
I mean, it's still better than nothing. So.
Oh, it's working now.
Oh, there we go.
Hi, Babak. How are you? Hi, Elena.
Hi, nice to meet you.
Nice to meet you.
James, can you hear us?
Hey, James, can you hear me?
I
Yes.
can hear you.
When I try to, oh, there it goes. Yeah, I was trying to start my video and it said I blocked assets, but it looks like it's working.
Hi, Vikram.
Hello.
Hi, everyone. Welcome back. So we're going to start the panel. We have moderator here in person, and we have a moderator Elena on Zoom, and they'll introduce themselves and the panel members.
Awesome, great. Thank you, Sarah, and thank you everyone for attending the industry panel. So today we have really five fantastic panelists who will introduce themselves a little bit. But I'm Ichin, as you know. I talked this morning a little bit about myself, but I really want this panel to be interactive. You can, we will ask some initial set of questions to the panelists over here and on Zoom, but we'll also have some time at the end for you to ask any questions you might have for our distinguished panelists as well. So with that, I will pass it on to Eli to start introducing themselves.
Thanks very much. Yeah, happy to be here. I mean, I guess by way of quick introduction, I know one of the first questions is sort of what path we took to get into industry. So I was originally a physicist in my PhD program and moved pretty quickly into computational biology. And what drew me in was a project that my advisor was involved in. The C4 Rice project, which some of you may be familiar with, which is this interdisciplinary effort to re-engineer the photosynthetic pathway of rice to increase crop yields. And so from very early on, I was interested in those kinds of multidisciplinary biological engineering projects. I was peripherally involved in another one in my postdoc. And I found that it's often quite awkward to do those things in academic contexts. And that's, you know, no knock on any of my collaborators. I was privileged to work with some of the best in the world in both plant science and synthetic biology. And their commitment to their projects is deep and sincere. But I think many of them would agree that you're often sort of swimming against the tide in attempting to pull people from different disciplines together and really, you know, make a new technology work in that framework. So I was very curious about how things looked from the industry side. Joined a microbiome startup in Cambridge as its third employee in 2017, and I've gone through a variety of positions in the industry ever since.
Hi, I'm Bora, and I'm a machine learning scientist at Shape. So a few years ago, I got my PhD here at UW, but I got it in electrical and computer engineering, and I was more focused on robotics. If you had told me at the time that I would end up at a gene therapy company, I wouldn't actually be surprised, to be quite honest, because my research was fairly unapplied. So after graduating, I worked at Amazon for a little bit over a year, and I kind of realized that probably wasn't a fit for me because although there was a lot of smart people there working on interesting things, I think the objective we were trying to optimize was just not a good use of time, perhaps. And, you know, potentially I was motivated by some extent because my girlfriend has a rare genetic eye disorder, and believe it or not, her eye disorder was, I think, the first one that was treated, our first successful gene therapy clinical trial. I think it's like called Luxtren or something like that. And I think that made me realize that gene therapy is a growing field and one that holds a lot of potential. And I think I just feel very lucky to be in this field right now. And I think I think that a lot of the times I tell people who are still in the more tech realm that feels like we're at the early days of the internet. I think it's going to play out a little bit slower, but I think we're figuring out how to reprogram ourselves. And I think it's going to have a lot of consequences. Hopefully a lot of them are good.
Shall we turn it over to the folks on Zoom?
Babak, you can begin.
Sure, thank you. So should I just introduce or answer the first question as well?
Maybe introduce and talk about how you got into industry, which I guess.
Sure. So my name is Babak Ali Panahi. I am the CSO of Excite Bio, not CTO. It's okay. So I've done my PhD in CS, but I started as an electrical engineer who was building robots, etc. And one day I decided that I don't like that field anymore. not to this, EE, but I retracted my admissions and I waited a year and applied to CS programs. And then I did my PhD with Mingley in Waterloo on protein structures, then moved to Toronto and, moved one step up in the dogma and started working on RNA with Brendan Fry and Ben Blanco. And the technology we created and the science became the foundation of a company called Deep Genomics, which does ASO therapies now. Continuing, going up the ladder, I joined 23andMe to work on genetics and AI models for disease risks. And then from there, I joined even one, step up and spend my time at Google Brain Genomics team on AI-based phenotyping. And, an idea that I see has got a lot of attention in the recent years. And after a couple of years at Google, I co-founded Excitebio with my all-time friend, Hani Gudarzi, who is a PI at UCSF. And, we are a liquid biopsy company using cell-free RNAs and AI to detect cancer early. So how did I get here? I would say, the best thing about industry for me is pace. I am not a patient person. I want to get results fast. And that's why, I like fast-paced environments that you can try many different ideas really quickly. Before going to, to the detail of my answer and, passing to Ikram, I will say that before leaving Google, around like 2020, I started a project, and that paper was published 3 years later. And some of the papers from projects that I started at Google, they haven't even been sent for review. I mean, I love it as a researcher. I love to have this on the side, but as a person, it kind of, the timelines are too long for the way I want to do things. I don't think, there is a right or wrong and there is any kind of preferred way. It's just, how people view and how they want to, go on with their research. And with that, I pass to Vikram.
Thank you. So first of all, I just want to thank the organizers for inviting me here. It's a pleasure to be part of this extraordinary meeting. My name is Vikram. Just to start with like my beginnings a little bit, I completed my PhD in David Bartel's lab at the Whitehead Institute at MIT. And I was there from about 2009 to 2015. And back then I was really interested in using machine learning approaches to predict microRNA targets, interested in post-transcriptional gene regulation field. And after graduating, I moved to Seattle for a while at the University of Washington. I wish I could have been there in person again to experience it again, but for now it's virtual. But back then I was a postdoc working with Jay Shinduri in the Genome Sciences department. And then it was around 2015 and been seeing news reports about deep learning, and that really caught my attention. But not that many people had applied that back then to genomics. Actually, Babak was one of the first papers I read in that area, so it heavily inspired my work. But that was kind of that time period of a relatively nascent application of deep learning to genomics, and it really caught my interest and attention. I became passionate about applying that to slightly different problem of that of transcriptional gene regulation. And then as time evolved, I started seeing just amazing work going on in industry as well, especially research projects coming out in the deep learning ML AI space from tech industry. And that kind of drew my attention a lot. And I was really fascinated by that. And also saw in biotech, there were companies emerging that were increasingly doing scientific research, like Calico, Illumina, for example. And this led me to kind of join Calico, and that's where I worked with David Kelly for many years and collaborators at Google and DeepMind for the next 3 or 4 years. One of my final projects at Calico before leaving had to do with a project to predict RNA degradation rates. And I would say it wasn't exactly the hottest topic. Not that many people cared about this very particular niche area. But it was during the time of COVID, and we really saw the blossoming of amazing technologies for mRNA therapeutics coming from Pfizer, BioNTech, and Moderna really blowing up this field and demonstrating RNA as a new class of therapy. So I got increasingly enthusiastic about applying some of my work into that direction. And just so happened that Sanofi was entering that market and created a new division dedicated to RNA therapeutics and research and development in that area. And the time and place was right, so that's where I ended up hopping back, and that's where I am right now, and leading a team of maybe 9 people or so in the ML RNA therapeutics space. So that's kind of the trajectory of my journey overall. Thanks.
So going on to the next question, some of you touched on this a bit in the introduction, but I'm curious, what has surprised you most about research in industry versus academia? And what do you think are specific areas that people are more well suited to do in either one or the other environment?
Does either of our in-person speaker wanna?
I can go for it, guys. So I think at least it's becoming probably to some extent increasingly clear that I think industry is well suited towards working on problems that require a large amount of engineering or engineering science. So like building large machine learning models or pursuing some type of novel biotechnology. But I do think that academia kind of retains a distinct edge in trying high risk kind of curiosity driven research, because a lot of the time I find in companies that, especially smaller ones, that you're working towards a goal, trying to like get something into the clinic. And so usually the problems you're tackling is you're trying to enable something to get it to work well enough to get to the point that it can meet some type of objective. And so I think these different aims kind of facilitate different types of research.
Anyone on Zoom want to chime in?
Look,
All right, no one on Zoom seems to be. Oh, sorry. Go ahead.
Oh, I was asking if either of you want to chime in if you didn't hear him.
Also on Zoom?
Yeah, if either of you.
Okay.
Oh, yeah. First time I went first, so this is you, Vikram. I will follow
Okay,
up.
yeah. So I would say, yeah, one of the most important and amazing lessons I've learned in industry is just how many different ways there are to break a product from practical success. So just want to give you a concrete example of as I work in the vaccine industry, there's some amazing papers in top tier journals showing universal vaccines, but then could they actually work? There are actually many different ways to break that. So just to give an example, you might need to refrigerate that material at very cold temperatures. You might need to ship it long distances. The dose might be too large, so it might be too reactogenic, might be too cytotoxic. So there are just so many myriad ways to break a product. And I would say being in industry has really expanded my thinking into all these different nuances of engineering, and you have to get all of them right to design the best product. For example, if you're working in developing nations, they may not have good electricity infrastructure to store vaccines at cold temperatures. So these are the kind of real world challenges that limit applied science applications, and has really pushed me to think more deeply about engineering solutions in those cases. But in the flip side, I would also say that where academia might have the advantage is that they might have the luxury to work on high risk, high reward projects that require more long-term rigorous work on time spans that may be unsuitable for industry. So that could be one advantage of academia versus industry, but ultimately both of them are very crucial towards advancing therapeutics and these types of products. Thanks.
Yeah. Okay, so my take is that first of all, I want to tell people that I think our similarities are far more pronounced than our differences. This is my first position. some of my friends, they joked, when I joined 23andMe that, oh, you are, moving to the dark side. There is no dark side, there is no light side. We are all scientists working toward, a really high level goal. And, we want to make life better. We are in computational biology and focused on AI in medicine. So for me, honestly, the first change that happened, and I try to complement the other panelists' answers, not to repeat what they said, which are amazing points. I remember the first Friday, someone told me Happy Friday, and I was kind of shocked. I was like, what do you mean Happy Friday? And it turns out that people actually celebrate Fridays because they have weekends. I had a very prolific postdoc, and I enjoyed it to the max, but I was working day and night. And, I started to appreciate evenings and weekends. And, I started reading books. I was, it was good to, continue my sci-fi reading a bit. So that was a great help. So not only for myself, but at places that I can make a change, I am trying to make that transition. So I am leading a team of like 30 scientists and engineers, and I really want them to have a lot of free time to, spend time on non-work activities. My view is that if you work 40 effective hours in industry and maybe in academia, you will be successful, and that's enough. Another factor, which maybe kind of alludes to the dark side, is compensation. I have to be honest with you, when I was a postdoc, life was hard. I was married and I had a kid, and you cannot deprive your kid, which means that you have to, cut from yourself and your well-being. And it was really hard. And I'm glad that at industry, I can, provide and create an environment that people can be comfortable. I mean, it's Bay Area and no one can, be well off. It's impossible, but at least, they can feel confident about, their financial situation. My view, my objective is that the people who I'm leading at any point in time, this is the best place for them. And I think in academia, it's a similar thing because, you are being trained, your compensation is not as high, but the reward, the long-term reward, could be really significant. But I think balancing it is hard. I think PIs in academia, they don't have as much leverage to reward, superstars or people who are really performing well, which is something that, I would love to hear from, my academic friends in future. But I think like in the industry, you have more ways to make sure that your team is taken care of, both in terms of like their mental well-being and their future prospects.
If you're done. I'll offer a couple of supplementary thoughts. I mean, I was really delighted to see some of the work earlier today on comparing the foundation model. I was really glad to see earlier today a lot of the work on really, kicking the tires on the foundation models and asking if they're outperforming much simpler approaches. And I think that's something that's often neglected in academia. And I think that perhaps surprisingly, industry can be a good place to do that kind of thing because it's really just quite sensible, to carefully evaluate the performance of a new technique on an independent test before you start actually betting your project on it in any way. And in particular, like if you can find a way to replace something complex and expensive with a $2 calculation involving logistic regression, that is a fantastic win. 95% of the time, the other 5% of the time being in cases where, you really want to be talking about the complexity and sophistication of your technology, which is a separate story. Similarly, I've had good experiences doing projects in industry, which, where there was an opportunity to boost performance by just doing a lot of skilled, difficult, technical work that wasn't particularly novel, right? And there's not a high-profile publication that comes out of that, but often there's a lot of payoff. And that's something that I think can often be done in industry if you can make the case for it. And that's something where I think industry can often support the community as well. I guess the final thing that I'll say, sort of the flip side of the compensation question, is like the attitude towards spending money, very different in industry. And not only in that the amounts that you're throwing around are often much larger, but if you're faced with doing a $500,000 experiment to get a critical result, the right response is often, okay, but if we spent 10% more, could we get that sooner? Right. And that's not, I think, how many of us ever thought in the academic space. And it is a real adjustment, right? It's quite challenging.
Great, awesome. So it sounds like our panel has really great views about how academia and industry share their similarities and differences across the research space. So the next question I have for the entire panel is, based on what you have I guess, worked on in both the research space in industry and academia, what are some of the best ways for academics to collaborate with their company? Both can be from the level of having an intern who work on research problems, but also extend to, say, for instance, a PI who's interested in leveraging some of the ideas and data that the company might be collecting.
I guess I'll go first. So actually, this is an interesting question. I saw a version of this when I was at Amazon. They had something called the Amazon Scholar, where they basically paid professors to come work there and consult. So that was one form I saw, and that's specifically tailored towards PIs. But that basically extends to postdocs and students because they could just go either intern or they had other programs that were like year-long fellowships. At a startup environment, it's harder to say. I think potentially there would be opportunities if there was some type of shared interest, some type of shared research. I know my advisor is currently funded by companies asking him to do some type of research that is useful to these companies. And then I would say like the most kind of natural collaboration I see on a day-to-day level for me is academic software. Anything that's put on GitHub and is used, I think, is kind of a very natural way to collaborate, and I think serves both aims very well, both academic and industrial.
I can go next. We have multiple active collaborations with academia. I love the way academia can do high risk, high reward projects, and that's the beauty of it. We have a couple of SRAs with a few labs. So these are service research agreements that you kind of have a contract with the lab, and they will do an experiment and share the results with you. I think these are very prolific because usually when there is like some finding from the study, there's going to be a joint publication. I think having interns in the company is great. It's a great way to see whether you like industry or not. I highly encourage, if there are any trainees, to do an internship because it's gonna be a surprise. So when we hire scientists, the first week they're usually taken aback by the level of rigor we have for coding. And the code is going to be reviewed, and the code needs to be replicable, and it should be run 100 years from now in the same manner and reproduce the exact same results. You might not like it. You might decide that you want something that is more researchy, if that's a term. But overall, I think we have never had any issues with PIs. The problem is usually universities and the legal offices that we need to figure out. I think they are stuck in the past. So sometimes, we want to use tools from labs, and these are tools that my friends have made. Sometimes these are tools that I have made, and I want to use them, and I have to negotiate with my own university, and it takes such a long time that we just give up. I hope that, we can make this simpler, easier to do. And I think there is a lot to be gained from collaborations between industry and academia, in particular for really futuristic high risk projects that usually are, the most rewarding.
All right, I guess I can go next. I would say that now I'm in industry, I started to realize that many of the academic partnerships that I've built between Sanofi and academic partners were people I met earlier in my life at some point. So these are from prior universities that I worked with or academic networks, people I met at conferences. And when I was a trainee, the concept of industry was quite foreign to me. I had no idea what people are really doing in industry. I would attend conferences. Academics would be sitting on one side, the industry people would be sitting on the other side, and we wouldn't really communicate that much. So I think it's really important to break down these barriers and start communicating across the space. And I think that's basically already kind of happening. The lines are getting a bit blurrier. There's a lot of cross-pollination of ideas between both communities, especially when it comes to data-driven ML or AI approaches, resculpting the landscape of what's possible therapeutically. So I would encourage people to make those partnerships and connections, because a lot of working relationship depends on trust and getting to know each other before even initiating the work. And I think if you see areas of opportunity, then by all means reach out to each other, and if there's a good connection, the industry partner would be more than happy to support and fund the work. But there are definitely a lot of complex legal and IP barriers, as Babak pointed out, and those in part are due to these university policies, etc. But hopefully we can forge a way through that as things evolve into the future. And just also want to mention the great opportunity as working as interns or co-ops, because when I was in my last year of PhD, I did an internship with James here actually in Microsoft Research, and that kind of opened my thinking into what life in industry is like as well. And now in this position, there are co-ops joining my team. We'll be opening up several positions in the summer and coming year. And I think those are other great opportunities if you've never experienced industry to get an inside glimpse into how things are done in a different environment. Thanks.
I don't think I have too much to add to this one in particular. I mean, I will reinforce that I think what deters, people in industry from doing academic partnerships is the question of time, which often really does come down to the necessary paperwork and working with universities. We have had some, I think, productive conversations where we've managed to set up pretty informal interactions along the lines of, hey, do you want to try this? With a minimum of like real transfer of either money or IP. But of course, that isn't always possible. Internships can be fantastic. It can also be a little bit hit or miss, even within a company with a good track record of supporting interns. So, if you're considering that path, you asked a lot of questions about exactly how your time is going to be used, and feel free to have a high standard for what you sign up to.
Well, yeah, it sounds like a lot of you have found ways to collaborate within industry and academia on joint projects and publications, which inherently leads to kind of shared research output. I'm curious if in other circumstances you found tensions between the company's internal need to optimize your particular platform or therapeutic NIP with a broader desire to publish and share your research progress. This could either happen because certain projects are inherently maybe more procedural or less novel, as I mentioned, but still very critical to the company. But maybe the more complicated case is when you do work that is potentially more novel, and it gives your company an edge, but then also it would be useful to share with others. But maybe for IP reasons or financial reasons, it makes sense to keep that internal. I'm curious if you've encountered that and how you balance that.
So I would say there's two main considerations. I think the first is protecting your IP. So before we publish or share anything, we have to go through essentially legal review at Shape. And that's fine. The lawyers are very responsive and very quick. But I'd say the bigger issue is time, because publishing requires doing well-controlled experiments, having good comparisons, nice figures, good writing. All that takes a lot of time and energy away from activities that are kind of more business critical at times. So there's always this tension between sharing and just getting the job done. And I think that will probably, based on my experience at Amazon, which is a very large company that has the market cornered, there you can kind of engage in more industrial research that's very, there's no immediate aim to it. But at a startup, I think the time energy constraint is like a limiting factor.
I guess I could go next. Oh, go ahead. Go ahead.
Okay. Yeah, I mean, the tension is real. I think there are, there is often a lot of appetite within the company, both the scientist level and often management level for sharing work. And there are a number of strategies, I think, focusing on like the software and machine learning areas for that. It's often possible to, keep internally at least a generation ahead of what you're disclosing. It's often possible to say, okay, well, we have sort of some infrastructure advances and we will share that, but we will not share sort of anything relating to our specific application areas within therapeutic development, strategies like that.
Yeah,
I'll go next. Oh, go ahead.
Sorry. I can say that, I have felt that in a really deep way. Industry is not only, rosy peachy. There are major issues as well. I discovered like some mechanism at a former company and I wasn't allowed to publish it. And 3 years later, it was published. And, to this day, I feel the pain. But, it's okay because, you win some, you lose some. But I always, try to open source as much as possible to make available as much as possible. But it's a tough battle, even when you are co-founder of a company, there are business interests and there are, other factors in play. So it's not an easy problem to solve. But my view on this problem is that you should always aim for openness, for transparency, for publishing, for sharing, as much as privacy laws permit and the non-scientific factors allow. But I think that should be the ultimate goal.
Yeah, I would also say that it's a very difficult and complex question that haunts me every day. But at the end of the day, I have to recognize as a business, we are a commercial enterprise, and to keep ourselves afloat and succeed, we need a little bit of a competitive advantage. So if a business is not selling, there are no sales, then the money dries up. If there's no money, then there's no research. So that's kind of a vicious, endless cycle. And year to year, there could be enthusiasm by venture capitalists, or the next year there might be a down economic market, and there could be a lot of squeeze and pressure on what you can make available. And so my philosophy, kind of like Babak's, is whenever possible to try to share my code in an open source way that's free for at least academic use. We do have to usually put a clause in there saying that it's not free for commercial use, but at least we can enhance reproducibility and gain more visibility and into what the technique or methodology was to kind of lift all boats, hopefully to progress the field. I would say arguably the hardest thing to do is share novel data because that data collection process takes a massive amount of human labor, monetary and time resources to generating those data sets. And so I guess one compromise so far that I've tried to do is that most of my life, my career as an academic was built on reanalyzing public data. That's been a big passion of mine. And I think public data, it's kind of still a goldmine of information. So that's something that I've still continued doing at Sanofi, trying to model public data. And the path to publishing on public data is a little bit easier than on private data. So that's kind of the compromise I've done at the present moment to still show the progress of our work to the world. Thanks.
Great. Fantastic answers all around. So for the next set of questions, I actually want to take a step back, and I'm sure many in the audience can echo with me with the sentiment that the machine learning for CombBio is really evolving really fast, right? I still remember my first ML class Transformer, where I don't know what this thing would be. And then now they're all over the place and people are using intro, I think even in the intro to like CS classes, people are using things like Hugging Face all over the place now. So I think for the panelists, my next question is going to center around what kind of machine learning skill sets, as well as biology knowledge, do you think most prepare someone who wanted to work your company or your field at?
So when we were graduating, they told us that our degrees had a half-life. And I think that's true now that I've been in industry, but I think the half-life on your various skills is different. And I've noticed there's one skill that seems to have extremely long half-life, and that's math. When I was here, I took this class, Mathematical Foundations of Systems Theory, which was basically an abstract linear algebra class. And this type of stuff just keeps popping up over and over again whenever I read papers and try to analyze them. And so I'd really recommend taking the time when you're in school, which I think is the best time to kind of beef up your rigorous skills.
I think that's a great answer. I'd go even broader and say that critical thinking, in general, is the thing that's never going to decay. And I think one thing that looks like in the machine learning field is specifically critical thinking about evaluation first. So I think that's a great kind of story to be able to tell in an interview setting where you have gone really the extra mile to understand what a model is and is not capable of doing. And I mean, I think the other thing, it's extreme caution about leaking information into the test set, right? That's the kind of thing where you can lecture people about it, but until you personally have been bitten by it, you're not going to have that like paranoia that you really need. And this is something that unfortunately, within the last 5 years in industry, I've seen a number of people, often not my coworkers, often instead people selling specialized services or consulting, make relatively elementary mistakes in this area. And so like the fundamentals, right? That's what's not going to decay.
Yeah, I mean, these are really great suggestions. I would say two things. First, invest in your coding. Try to be really better coders. Many of us haven't had formal software engineering training. Try to, learn some of the design concepts. It's going to pay dividends. It's going to help you so much later in your career. Try to write tests for your code. If you have a lab mate, ask them to review your code, give you comments, read other people's code. I think is the best way to learn, the concepts. Also, ML is AI ML. It can be a dangerous tool. I think the bane of our existence is batch effects these days. I've seen so many big companies go under because someone forgot to correct for one random batch effect. We had a poster yesterday on it. It's something that we pay a lot of attention. Okay, marketing wants me to call it confounder effects or whatever, but you know what I mean. When you get data from multiple sources, these models are amazingly rich and complex, and they can hone on them and they can learn them in a really, really malicious way. And the thing is that we would never know. I think, one of the panelists alluded to data leakage from training to testing. One thing we do, for example, at XI, is that the person who develops the model, they don't have access to the test data at all. And validation data, they have a budget. It's like Kaggle. So, they can run validation data maybe 5 times. There is a budget that we decide before starting the project. And, I will finish with this. Pay the proper respect to the baseline models. A lot of times, I have been applying deep learning for many years to genomics, but I am the first person that will start with a linear model or with a proper baseline. A lot of times good baseline models, really work well. I think there was a paper I saw that compared linear models with the new foundation models on single cell data and showed the linear models in some applications were actually better or I think in most applications, we need to be really careful about this. And as always, a good AUC is just a beginning. You have to show that you have earned it and your model really, is doing the proper job and is giving you a bona fide performance.
Great. Yeah, I'll add to that the type of team that I'm trying to cultivate is people who have dual expertise, that they have scientific domain knowledge coupled to ML and AI talent. And in my mind, it's not good to have only one or the other. There needs to be at least a little bit of both. And by domain knowledge, I mean deeper knowledge about mechanistic biology, gene regulation, molecular biology, RNA biology, classic bioinformatics and cheminformatics. But in terms of ML skills, would also echo what several of the other panelists have said about the fundamentals of classical machine learning techniques, rather than just taking a class on the top of the line AI or deep learning. There's a lot of hype surrounding these, but oftentimes in many scenarios, as people have said and shown in several great talks earlier today and yesterday, these simpler methods often have many advantages in usability, interpretability, etc. So I would resist the urge to train yourself only in the advanced techniques. And in many real world scenarios, such as data limited ones, they might actually perform more poorly, as several people have mentioned. So the domain knowledge is also critical in defining the metrics for performance and understanding the nuances of the data, such as how reproducible the data is. Are the error bars very large on the measurement? What are the limits of prediction? So you don't just get in that machine learning mindset that if I improve my R squared by 0.01, that is a breakthrough. Sometimes that doesn't matter. And sometimes maybe you're already training a pretty good model. So that is a little bit of not just a science, but an art form, how to approach those types of questions. So the domain knowledge will help guide you a little bit into that art form of designing the exact type of benchmarking that actually matters. Thanks.
Yeah, everyone's gone. Those were all really interesting and helpful answers, and also point to some of the skills and challenges that get emphasized a bit more in certain industry contexts where the focus is really about using the models you're working with more than just trying to get certain performance. So our very last question before we get to questions from the audience are, given all of this, what are the research areas that you're most excited about developing in the next 5 to 10 years, whether you're working on it yourself or have just kind of seen it bubbling up in talks over the past couple of days?
Yeah, sure. I'll jump in real quick. I mean, so my current company, as well as my previous one, I've been working on drugs to increase gene expression, and the first area of application for that is typically haploinsufficiency diseases, patients who lack, have only one instead of 2 functional copies of a gene. And similarly, with so many of the really advanced technologies around gene editing, around mRNA therapeutics, we're in the early stage of the development of those technologies, and it's quite difficult to get them to work. So you often want to use them to do something fairly obvious, right? In the sense of, if you can get it to work, it will correct the problem. But there's going to be a next generation after that, which is, okay, well, if we have these general purpose tools, how do we go in and do completely non-obvious and unexpected things to treat more complex diseases? And I think that modeling is going to have a huge role to play in figuring out what the correct way is, or what the best way is to approach those problems. Don't know exactly what that's going to look like, but I think we have seen a number of talks in the past couple of days that sort of start, moving in the direction of really capturing much more complex processes at larger and more systematic scale. And I think that's going to be fascinating.
I mean, I work in the gene therapy space. That's probably obvious, but I think the one that it's really delighted me recently has been developer tools, or more specifically, ChatGPT and GitHub Copilot. I think GitHub Copilot has let me not focus on syntax as much and just focus kind of on the ideas when coding. And ChatGPT has been amazing for firing off emails that I didn't want to write, in addition to analyzing papers and like working through some math. And so I think as those continue to advance, I'm looking forward to doing less of the mundane work and focusing on more interesting work when I'm in my job.
Yeah, So, let me talk about something, maybe unpopular. Everyone is looking for LLMs that can answer questions. They are useful, but I think we need to think about an LLM that reads our papers and asks questions instead of answering them. And it should ask questions that probably we couldn't think about. I hope that we can build them. My confidence in, for example, ChatGPT is periodic. Sometimes it gives a really elegant answer, and sometimes for very simple questions it flutters. But I would say I have been working and thinking a kind of a knowledge base, a system that can start by answering the major questions I have, because for every question you have to start searching. And I think a general GPT-like system wouldn't be good enough, because they really need to understand all these new papers and be updated. But I'm looking forward to the day that we have this question asking AIs.
Great. For me, it's a little bit related to what Babak was mentioning, but I'll try to take it slightly different direction. So probably many of us have been looking at the OpenAI drama playing out over the past few weeks, but one topic in my mind that evoked was kind of like the holy grail of the AI field for the last 60 or plus years. Everyone has been kind of trying to define, is it possible to make an artificial general intelligence? And I can't say that I'm working on this, but it's something I have my eye on very keenly, just to see that, how do I define AGI? Something that an AI that can reason, perceive the world, ingest large knowledge bases, not just use AI for pattern recognition with large data sets, but something that can actually reason and learn causal or mechanistic principles to generalize to out-of-domain scenarios. And that's kind of a struggle that several people in this conference were talking about, how to leach something to actually generalize to those OOD scenarios without being as data hungry as current systems tend to be. So to me, this particular space is really fascinating, and not just AGI, but really the confluence of it with computational biology as well. We're seeing an era where there's mountains of public and private biological data growing exponentially year by year. And how do we actually make sense of that? Maybe an AI, like an AGI or something like that, if there is a breakthrough in the field, it could actually make sense of that and learn causal principles. And I view that as possibly, even if we don't invent AGI in the near future, the research going into that direction might still give influence into engineering novel proteins, small molecules, nucleic acid-based therapies that are still extremely valuable. So that to me is my interest in the next 5 or 10 years, just seeing the interface between AGI and the comp bio or therapeutics field. And so it's a very exciting time to be part of that and contribute just a little piece to it. Thanks.
Awesome, great. Again, just fantastic answers all around the panel. With that, that concludes our prepared questions. So we would love to open up the floor to any of the audience who have a question for our panelists.
Hi, everyone. So I'm a neurobiologist and a clinician, and recently got more interested in and also bioinformatics for a long time. But I'm interested in how you guys see biology background, machine learning background. Both are very technical, can take a lot of time to understand, and they're getting increasingly more complex over time. In your spheres of people you work with in your company, is it very divided? How is it split? Do you feel confident that that it's balanced and people are understanding both sides?
I can take this. I think there was a time that this process was torturous. You had to. I didn't have good biology background, so I had to take a lot of courses. and do a lot of independent study. But I think the new generation of scientists, like they are really they are very good coders, very good scientists at the same time, covering machine learning, biology, for example, many students from the labs of organizers of MLCB. So I think the next generation, or I should say current generation, they are doing really well. And honestly, I think you cannot have disjoint groups. That's why I am favoring self-sustained teams, as I call them. So for example, in my own team, engineers, computational experimental biologists are under one umbrella. We actually, before this, we had our weekly meeting, and I asked the team to help educate and elevate the level of competency of the entire team. A software engineer who's spending his time on bioinformatics and on computational biology, they should know, at least to some degree, what does RNA-seq mean and what are the limitations. Likewise, a biologist should know why implementing a simple change in the pipeline, why it initiates this cascade of operations and cascade of tests. So that has been my solution, to have a group of people that are working closely together and have shared objectives.
I guess I could go next from the perspective of being in a mega corporation, big pharma, as opposed to some of these other panelists. So in such a big organization, we have large teams of people dedicated to experimental or wet lab science. And then on the complete opposite side of that spectrum, people who are all AI, ML, pure digital. And then there's teams like mine that are kind of somewhere in between, trying to bridge that gap, communicate with both communities. And the biggest success is if all three of these functions are working harmoniously together. If they're working in an insular way, the experimentalists don't necessarily think in a very numerical optimization type fashion. And on the other end of the spectrum, if you're a pure AI, you might not understand the nuances of the data, where it's limited, these batch effects and other types of problems that might be prevalent in the data sets. So we need to all be speaking and communicating on a regular cadence, and try to digitalize all the information being produced and store that in hard drives in a way that's structured and organized, so that data engineers can harvest that information, and then people in machine learning community can actually analyze it and make sense of it. So I think it's really important that all these communities get integrated as much as possible for the success of the business. Thanks.
Yes.
So my question is about I think because I work with sequence-based deep learning models, so it's around that area. So I think so far the models that we have had, the large case models that it's not possible for academia to train, like Informer, like Borzoi. So they have been the weights and everything have been made publicly available, the training data. But if you think of the next generation of the models that will be trained, for example, something like big RNA, I think the model weights are not publicly available, right? So why I think my question is if we are moving towards that trend where we don't make these model weights publicly available, how would people from academia verify whether there's something wrong in the model weights, and or whether they're biased towards certain things and stuff like that? And as people from industry, being in the leading position, can you explain why is it difficult to make your models publicly available? Because you can literally put an embargo on that, okay, others cannot use it for commercial reasons. So why is it difficult to do that?
Let me just jump in with a quick point of clarification for big RNA. Although we did not release the code of the weights with the preprint, we are in the process of submitting that. The team is actually working on revisions right now to a journal for formal publication. And we do expect that there will be additional disclosure along with that. I don't know exactly what form it will take. With the emphasis that I'm speaking personally for this next part and not necessarily on behalf of TG Management or the big RNA project team, I do think that it is necessary to hold the line on requiring disclosure as much as possible. And I think, you know, a lot of the folks in this room from the academic and editorial side do have a big role to play in maintaining that community norm. Right? I think you're quite correct. You know, so often without like the fundamental standard does need to be genuine reproducibility, and without a disclosure of the model in the context of publication, that can happen. Now the question becomes more, okay, well, what happens, you know, if we end up in a framework where people are training these models and then not going the route of traditional publication? And I don't know. I'd love to hear from my co-panelists on, you know, what a good model for the future might look like.
So I would like to add something like, you know, it would not be possible for us to like, you know, build something and then compare against these models, because I wouldn't be able to make predictions on, let's say, a certain set of sequences we design and its variants, right? So I think it would become just impossible to know whether any model that anyone has built, whether it has built like it beats whether something like big RNA or like Informer 5.0 or something like that.
Amen. Right, that's an important challenge, right? It's important that that capability be preserved. Anyway, I do want to let my co-panelists speak.
Well, for myself, I was involved in Informer and Borzoi. We tried our hardest to release all that information publicly. At Sanofi, it took a few months of legal review, but finally yesterday we released our codon BERT model. So we're trying our hardest to release all these models openly, freely for the academic community. We just try to put that little tag in the license for a non-commercial use to try to protect our commercial interests. But other than that, it could at least advance the academic literature. But some of these are big legal gray areas. So imagine making a model that's so amazing it could make your company $1 billion. Maybe it's not the best to release that model immediately. It might be great for academics and for the world, but it would be a very bad business maneuver for the company. So they need to protect their commercial interest a little bit. And even if you do release the model under that non-commercial clause, this legal gray space makes it very difficult to validate or verify if a competing company is downloading and using your code on a regular basis. So I've been talking to different legal experts, and that even people who are legal experts in the digital space find this a very gray area. Like no one really knows how to protect these types of models once they're unleashed to the world. And I think the tech community is facing something maybe a little bit similar. Google, for example, invented the transformer and other types of technologies like this, but then their competitors like OpenAI are now using similar technologies and competing against the big fish in the tech world. So it's a very complex space, and there needs to be some sort of balance between these two, I guess, and it's not always easy to define.
Yeah, and a lot of times, the privacy issues. For example, my former team at Google, they made this protein annotation, platform, and they made it public. It's now part of, UniRef, etc., UniProt. But the thing is that, let's say we build a foundational model based on, RNA profile of, 10,000 cancer patients. So can we make this public? I mean, how can we guarantee that it cannot end in identification of patients in our cohort? We had the same issue at 23andMe, for example. How can you make sure that you are preserving privacy of participants and you are true to, the consent in the IRB? I don't know. It's like over my head. I think these are really complicated questions. But I've been thinking, maybe we can find middle ground solutions until, lawyers can solve it. For example, I think at Google, we had this option that, maybe we cannot share this model, but we have an automated system that will, apply this model to your data. And it's gonna, guarantee your privacy. It's gonna, run in a independent, container. Maybe we can provide, access like that. But obviously, if we can find a solution, we're gonna, seriously consider it. And if it works for us, definitely embrace it.
Great, fantastic answers all around. I don't know what to say. We can probably go one more questions given the time limit. Anyone want to do the honor of last questions from the live audience?
Thanks for all the great discussion. Something I've been thinking about recently of a difference between academia and industry is how collaborations between computational and experimental groups work. So, I haven't been in my faculty position that long, and I've already had this issue of what was overall a very successful collaboration with an experimental group, but we still ended up having this, I would call it a discussion, but it was, I think, technically an argument about who should be first and who should be last, because we felt like, the computational components were the main contribution to the paper. The experimental collaborators felt like it was the data generation. And so I think this basically comes down to, in academia, our careers are primarily judged by publishing papers. Papers have authorship lists. And somewhat, and that's just that's not something that's going to change, unfortunately. Maybe there's better ways that we would ideally assign credit of who did what. But yeah, I'm curious how you feel, whether that's something that you feel is a substantial difference, that because in industry your goal is getting to some product, getting to some therapy, and so it's not about who was last or first on a paper.
I mean, I can, so that's actually a nice thing about working in industry. Maybe the names on a paper really don't matter that much, but I think in terms of how we structure our projects and how we make decisions, there is definitely a tension between experimentalists and computational folks. And I'd say a big part of that is having just a mutual respect and having people on the experimental side that understand what are the benefits of bringing the computational folks, and I think vice versa. And I think that's one thing at Shape that we wrestle with, and I think we're actually pretty good at, is collaborating very tightly. So that means going to meetings together, talking, and not just sitting in separate siloed parts of the company, because
yeah, I mean, paper authorship is less of an issue, but you can substitute in, I think, the separate question of respect, or more practically, who's really having their voice heard and who feels like their contributions are actually being well used, right? And in some ways that becomes even keener, right? Because it's okay, if you're in a situation where your organization is dominated by people who come from the other side of that divide, and they walk away from an opportunity and leave money on the table, or they have unrealistic expectations for what they need you to do, and it could have gone differently, and ultimately a project fails, a company fails, probabilistically, it changes the outcome for patients. It's a very upsetting experience. And I've worked in companies which were sort of more from the classical biotech mold, where the leadership had that kind of background. And in those cases, it can be quite difficult to educate people on what's realistic from a computational perspective. At Deep Genomics, we really came more from a machine learning setting, and the shoe has sometimes been on the other foot, where the biologists have things to contribute that we need to make sure actually really get taken in. So I mean, I guess the short answer is that you do see the same problem in a different guise, and I wish I could say that that didn't occur, but it's one of the challenges of doing work in this space, however it appears.
Was it David who asked that question? I can't.
Yeah, I was. Hey, Babak, good to see you.
Yeah. So, let me tell you a funny story. So we had a collaboration with an academic institution, and they sent us the draft, and we were somewhere in middle, and we didn't even have names. We were like exi author. I'm kind of relieved that I'm not fighting those fights anymore, but at least, when we collaborate, it doesn't matter that much for us, except, sometimes you have younger trainees who really want to, stay kind of in the publishing pool. I don't know. I think this is an old model. I wish we had a better way, because like these projects that we are doing, like some of them take 5 years. They are long-term projects. There are many people who are contributing a lot, and it's a bit unfair to kind of assign credit to who happened to be physically first or physically last, or as David calls them, first or last. But I'm glad that it has been less of a concern. And if you ever work with us, I can guarantee that we are very easygoing, and we usually make sure that our academic friends are happy.
Yeah, I would say same for me, that overall authorship has started to matter less to me than it used to, but I just still try to attribute credit where credit's due. Like the people who've spent a lot of time and energy and resources into it, they should be first and last, and when possible, share authorship as well. But that problem has been a problem since the dawn of time, even in academia, no matter where you are. There are many times in academia I was dissatisfied with my authorship, but the cards didn't quite play in my favor. But I would say arguably more important than authorship is just the protection of commercial interests and IP. And I think that that's a little bit more of a complex area. And as Babak alluded to earlier, there are many times when I'm extremely aligned with a professor in a different university, but then you get all the lawyers involved and the legal teams aren't quite aligned. And then something that's supposed to be simple quickly becomes much more complex than it started off with. So the organizational structure can contribute a lot to this complex IP discussion. Thanks.
Great. With that, I want to give us our distinguished panelists a round of applause. And I'm sure they're happy to be reached, and you are feel to grab them when they're here in person or online via emails and other messages.
Thank you.
Thank you.
Have a nice thank you, everybody. Thank you, the panelists. Oh, sorry, Vikram, was that you?
Yeah, I was just saying thank you. Appreciate it.
Thank you all. So this concludes MLCB for this year. Thank you for fantastic submissions, fantastic talks, fantastic questions. It went very well. And just one thing before you leave, if there's trash on your table, please throw those out so that we don't have to stay after hours here. David, anything?
Yeah, we will follow up with a survey. We'd love to get your thoughts on various things, including venue for next year. I think we have a choice of doing here or New York, so we need to figure out what people's preference would be. So, yeah.